Hello students, welcome to Vedantu NEET English. I am Ms. Gopika, your biology master teacher. I hope I am visible, audible and the presentation right behind me is visible. Please let me know in the chats if I am audible, visible and are we live yet? Yes students, are we live? Not yet? Yes, I think we are live, right? Right. Chalo, we are live. Awesome. My health is better students. Thank you so much for asking. It's doing better. So today, depending on the class, my health will get more better, right? If you understand all the concept, then I'll be more happy. And happiness is equal to giving good health. Okay, so today we are going to do molecular basis of inheritance, one of the longest chapters. And my students cry telling that, ma'am, I cannot read NCERT. So I thought, okay, why not? I'm there, okay, reading NCERT, teaching NCERT, so I'll be teaching you every single line of NCERT. And students, this chapter is one of the highest weightage chapters. Highest weightage chapters means how much, ma'am? 5 marks, 5 questions, 6 questions? No, around 8 to 11 questions almost every single year repeats from this question. That means every single year, if you go and check my weightage chapter, where I've put the weightage of each chapters, you'll be able to notice that this chapter all the time, okay, every single year, it fetches us easily, around 8 to 10 questions. Okay, that means around 36 to 40 marks from this chapter. Okay, up to 44, 45 it will go. Okay, so it's a very important chapter. So quickly smash the like button. Those who haven't smashed the like button yet, mark your attendance so I know that you were present, and it makes me happy to see your likes on the video. Okay, so do not forget, just now, right now itself, mark your attendance. Okay students, so good morning to all of you. Let's start this day on a beautiful note. Let's start with molecular basis of inheritance. So we are going to divide this class into 2 parts, because this chapter, if I take a marathon, it will take around 7 hours. 7 hours I can take a marathon on this chapter, because it's that long of a chapter. So we'll divide it into 2 pieces. Okay, so the first piece today is we are going to learn what is nucleic acids. Okay, what is everything about your DNA and RNA, then structure of your polynucleotide chain, how they are formed, right? How do they assemble themselves to form the entire polynucleotide chain, then the experiments, how did they prove that DNA was the genetic material? Chumma, they didn't come and stand and be like, oh, DNA is genetic material. No, they were experiments to prove that DNA was the genetic material. And then replication, everything about your DNA replication. See, subtopics I haven't added, but you will learn a lot of subtopics in between these topics. Okay, so are we ready? Are we ready, all of you? Is the energy high? Is the morning going good? Awesome. Yes, I think Gracie, Pavitra, Meghana, Atuluru. Yeah, I think I'm pronouncing the name wrong. Okay. Yes, Jay, Madhumati, awesome. The energy is very nice. Okay, chalo, let's start the class. All of you smash the like button and mark your attendance, and we're going to start the class. Okay, now students, before I get into the whole chapter, now what is this chapter about? See, all organisms need someone to store all the information, right? To make them see, sometimes when we go see a baby, right, a just born baby, you'll be like, oh my god, the baby looks exactly like their mom, looks exactly like their dad. Now, how do this happen? It is because there's something to store all the information from your paternal and the maternal side, and that protects it so well and carries those traits to the next generation. Right? You will all think, oh, my mom has such a good hair, but I haven't got any of it. Right? Because some of the characters will be like your paternal, some of it will be maternal. Sometimes both mixture, you will get one type of character. Right? So all the molecules or all the organisms has a set of information. All of you are following me so far? Yes? Yes or no? So a molecule that has all the information, students, a molecule that has all the information. Is there any friend of yours who knows everything about you? I'm sure no one will be there who knows everything about you, right? But there is a molecule that has all the information, okay, of all the organisms, okay, of all the organisms. Of all the organisms. You do not have to write the notes. Okay, you do not have to write the notes. You will get this notes on Vedantu NEET English Telegram, as well as if you want to be updated about my classes and some about my life, then you can definitely join Biology by Gopika Telegram right now itself. Okay, and yes, That's it. So students, a molecule that has all the information of all the organisms. Okay, no matter what, paternal, maternal, every information is there. But can I say, put a statement telling that all the organisms have nucleic acid? Yes, ma'am, you can definitely tell that. All the organisms have nucleic acid. Yes, agreed. Right, all organisms have nucleic acid. But who is this nucleic acid? Why are you talking randomly? Nucleic acid is who, students? DNA and RNA. I told all the organisms have nucleic acid. That means almost 99.9% of your organisms have DNA as the genetic material, and that rest of them, like your virus, your tobacco mosaic virus, have RNA as the genetic material. So I can say all the organisms have nucleic acid because nucleic acid is DNA and RNA. Anyone disagrees to this? No, ma'am, we do not disagree to this, right? So DNA means what, students? Simple. DNA means deoxyribonucleic acid. What is it called? Deoxyribonucleic acid. Deoxyribonucleic acid. So what will RNA be? All of you in the chats, what will RNA be? Yes, very good. DNA, ma'am. RNA, ma'am. Very good. Now what is RNA going to be? Ribonucleic acid. Right? Ribonucleic acid. Now I told you majority of the organisms, right? Majority of the organisms are going to have DNA as the genetic material. No doubt in this. Very good. Okay. But other people, some few people are there who are little exception. They are like, I don't want DNA as the genetic material. That is your some viruses. Okay. Some tobacco mosaic virus. Okay. These are the ones who have RNA as the genetic material. But all organisms have nucleic acid as the genetic material. Assertion, reason question can come, statement based question can come. So you should be aware of this. You should be aware of this, telling that, okay ma'am, all organisms have nucleic acid. I agree to it. Okay, that will be DNA, RNA. But students, just by looking at the name, what do you understand? That something is missing, something is there, right? Look here, this is deoxyribonucleic acid. That means one oxygen got deleted. Here it is ribonucleic acid. Yes. So in RNA, one oxygen is present on your 2' end, but that oxygen is not there in DNA. Are you able to understand? So here one oxygen is less. Okay, here one oxygen is less. One oxygen group is not present. Does that make that much of a difference? Yes, it makes a major difference. That is because this one oxygen is not present, right? Because this one oxygen is not present, students, their stability when I compare with your RNA is absolutely high, right? It's absolutely high. Just because of that one oxygen that is not present, what happened? The stability has increased because oxygen is going to react with everyone. We have studied that, right? Oxygen goes and reacts with everyone. So oxygen will react because that oxygen is not there. Their stability has increased and the reactivity is comparatively less. But because these people have one oxygen more, what has happened? Okay, here oxygen is present. Okay, oxygen is present. Because of that, here reactivity, okay, here reactivity increased, not stability, right? Reactivity increased and automatically stability decreased. Could this be one of the reasons why 99.9% organisms have DNA but only few of them have RNA? Yes, ma'am, agreed. Because if I am not stable, see, if you are not stable, then there are chances that you take this decision also, you take this decision also, you take this decision also. That, okay, I will join batch there also, I'll join batch here also, I'll join batch here also, I'll study from this teacher also, I'll study from this teacher also. So you will be all around the place, right? So we do not want a genetic material like that. We want a genetic material who is like, okay, solid, like solid genetic material. Okay, so far understood, clear? Sorry, so far did you understand? So far understood, clear? Students, brain is not braining in the morning. Yes, very good. Did you all mark your attendance? Very good. I think all of them have marked their attendance. Okay. Now students, this DNA, RNA, they are made up of some chain, right? Everything has a component, right? Now if I have to construct a building, I will have lot of bricks. Many bricks together, I'll put cement and then I'll build the foundation. Same way, this DNA and RNA also have this brick kind of component. Can anyone tell me what is that brick? What is that brick? Oh, Sanvi is there in the class. Hi Sanvi. Okay, what is that brick, students? Can I call it nucleotide? Yes, ma'am, you call it nucleotide. You call whatever you want. Okay, I am least bothered. No? Others? Okay. Hasini is agreeing, Savita is agreeing. No, ma'am. Nucleotide. So, nucleotide, many nucleotide together will form a polynucleotide chain. Right? Many nucleotide together will form a polynucleotide chain. So, my nucleic acid, right? Let's say nucleic acid. This nucleic acid, wait, we'll write it here, students. Here, this nucleic acid is a polymer of nucleotide. This is a polymer of nucleotide. Yes or no? Now, what is this nucleotide? You never told what is this nucleotide. So, nucleotide is formed out of 3 components. What are those 3 components, students? What are those 3 components? First component is nitrogenous base. Okay, first component is a nitrogenous base. One is a sugar and one is a phosphate group. One is a phosphate group. I don't think any student will have doubt in this. Anyone who's studying this chapter for the first time? Anyone who's studying this chapter for the first time? Yes, very good. NB, sugar and phosphate. Nitrogenous base, sugar and phosphate. These 3 components are the components of this nucleotide. But is it only one nucleotide? No. Many nucleotides, okay? Many nucleotides combine together to form a polynucleotide chain. Many of them, okay? Many of them. So, this is going to form what? A polynucleotide chain. This is going to form a polynucleotide, right? Poly means many. Poly means many. Many nucleotide. Agreed? Yes, all of you agree? Yes, ma'am. Okay. Now, let's pick one. Okay, let's pick one, students. Let's start with sugar. Okay, even though I wrote here nitrogenous base, let's start with sugar. Now, what is this sugar? The one we put in the tea and drink? No. This sugar is going to be a pentose sugar. This sugar is going to be a pentose sugar. That means what? 5 carbon, right? 5 carbon. Since I haven't studied in my life so much chemistry as much as this chapter has made me study. Okay, 5 carbon. Okay, pentose is 5 carbon. Now, in DNA and RNA, there are 2 types of, right? 2 types are there. 2 types of sugar. One is going to be deoxyribose sugar. Deoxyribose sugar. One is going to be ribose sugar. Okay, one is going to be ribose sugar. Now, what is the difference? I'm telling deoxy, I'm telling ribose, right? Yes. So, here deoxyribose sugar, ribose sugar. So, I told you deoxy, that means one oxygen is not there. So, let's draw the structure. Okay. My God, bless me, students. Oh, perfect. Okay, here also let's draw. Yes, I drew a phi ring. Okay, I drew a phi ring. Now, let me label it. Okay, let me label it. So, students, here, let's say, okay, I'm just drawing this. You people will know better, but yeah. See, so we are going to label it, okay. So, this is going to be the first carbon. Okay, this is going to be the first carbon, second carbon, third carbon, fourth carbon. Ma'am, where is the 5th carbon? Hello, 5th carbon will be here, CH2OH. This is going to be the 5th carbon. Agreed, all of you? Agreed, all of you? Are you in the DNA batch? Then you would already know when we are completing the syllabus, okay? Yes? So, students, 1, 2, 3, 4 and 5. Yes? Same thing goes here. 1, 2, 3, 4. Then here it is going to be CH2OH. That is going to be the fifth carbon. It's going to be the fifth carbon. Done. Now students, look here. In the second carbon, something is going to be there, something is not going to be there. Okay, something is going to be there, something is not going to be there. Who is that something? Here H is going to be there, here OH is going to be there. Here H is going to be there, here OH is going to be there. Because of this O, reactivity has increased, stability has decreased. Are you all clear on this? So we have a 5 carbon ring, right? Pentose means 5 carbon. Pentose, okay, pentose means 5 carbon. So we have two types of sugar, deoxyribose sugar, ribose sugar. In deoxyribose sugar, as the name suggests, one oxygen is coming less, okay, that is on the second carbon. Here one oxygen is more in the second carbon, okay. Oh, okay, Daya, nice, nice. Okay, yes. Yeah, some of you haven't marked the attendance. All of you mark your attendance right now. So much conjuice you people have become, okay? What, you people are paying money or what? To mark attendance. All of you mark your attendance. Yes, students, so far good. All of you look at the diagram and tell me any doubts? Any doubts? Sugar is clear? Yes, ma'am, sugar is clear. You made us understand that there is a deoxy group, that there is a ribose group, and on the second carbon, this is going to be an hydrogen, this is going to be an oxygen. Done. Okay, next whom we have to take? Okay, sugar is done. Next, we have to go to the phosphate group and nitrogenous base. Shall we go to the next page? Okay, next page, we are going to take up your nitrogenous base. This is a little complicated, a lot of chemistry, okay? Students, nitrogenous base. From the name itself, what do you understand? Students, today biomolecules class is there. That time you will understand the structures little more clear. Now you would have understood the foundation. In sir's class, you will understand little more detail. Okay, so it will be like full balance you will have. Okay, so students, nitrogenous base. So this nitrogenous base, especially your nitrogen, all of you know there is a heterocyclic ring, right? Heterocyclic ring, your benzene, all that you study, no? So it's going to be a heterocyclic ring. That is a ring-like structure will be seen here, okay? Ring-like structure. Now if I want to split, or I want to know who is coming exactly in your nitrogenous base, who is going to come, all of you tell me in the chats, who are the two people who is going to come? P and P. Very good, Hasini. Purines and pyrimidines, okay? These are the two people who are going to come. Who? Purines and pyrimidines. So here, let's say purines. Students, purines, as the name is small, but the ring is big, okay? These purines are actually a double ring structure. They have around 9, okay, 9 if you count carbons, no, 9 carbons will be there. Purine, small name, but 9 rings are there. Pyrimidine, big name, but only 6 are there. Okay, 6 are there. Okay, clear? Clear? Okay, I'm writing it here. It's a double ring structure. Okay, it's a double ring structure. It is a 9-membered. Okay, it is a 9-membered ring. You will understand when I draw it. Okay, 9-membered ring, and they have nitrogen. Very important question. Okay, they have nitrogen in 1, 3, 7 and in the 9th carbon. Okay, these are the places where they will have nitrogen in the 9th position, not carbon, position. Shall we draw this? Shall we draw? Gopika Ma'am is going to use all the chemistry that she knows in her life. Okay, I'm drawing it, students. Let's draw. 1, 2, 3. Okay, 6-ring structure. Perfect. I drew a ring. So we are going to count now, okay, if I drew it correctly or wrong. Okay, so here let's say we start from here. 1, 2, 3, 4. No. cyclic ring is wrong. All of you draw this first, then I'll start numbering. Okay, then I'll start numbering. You should start from here. Okay, not from here. Here from here. 1, 2, 3, 4, 5, 6, 7, 8, 9. All of you agree it's a 9-ring structure? Yes, ma'am, agreed, because you drew and you only labeled 9. Okay, so 1, 2, 3, 4, 5, 6, 7, 8, 9. Done? Done? Okay, so here you have cyclic and non-cyclic, but I don't know how much you will be able to comprehend. But the one that is forming the bond is going to move like this. Okay, the one that is going to form the bond, that means the one that is going to form the bond with the sugar. All of you know 1 and 9 is going to form a glycosidic linkage, which I'll teach you slowly. So students, this is a purine ring. Now where did I tell you nitrogens are present? I told you that nitrogens are present in the 1, okay, 1 first position, okay, in the 3rd position, right, in the 7th position, in the 9th position. Yes, nitrogens are present all these places. 1, 3, 7, 9. Okay, done. Done. Now we have to talk about the next person. Who is that next person going to be? Purine, ma'am. In nitrogenous base, the purine is done. Next is going to be a pyrimidine. Okay, pyrimidine. Now we are only studying we are going to learn the structure. After that, I'll tell you who comes under purine, who is going to come under pyrimidine. Okay. Something is wrong with the spelling, no students? Pyramidine. Okay, pyramidine. Okay, pyramidine. As I told you, name is big, but the structure is very small. Okay, Pavam boy this fellow is. The structure is very small. The structure is going to be a 6-ring structure. Okay, it's a single ring. Okay, it's a single ring. How many people are there? 6-membered family there, okay? 6-membered. And where will they have the nitrogen present? On the 1st and the 3rd position. On the 1st and the 3rd position they will have. Okay, shall we draw the structure? Yes, ma'am, you draw the structure. So it's going to be simple, students, a 6-ring structure, a cyclic 6-ring structure. Done? All of you, did you draw this? Students, you have to draw it. Okay, you have to draw it. Otherwise, you will not understand. Okay, so this is also going to be this way around. Okay, this way around. So I'll start labeling from here. Okay, I'll start labeling from here. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Yeah, 1, 2, 3, 4, 5, 6. Done? Okay, done. Okay, so here where is the nitrogen groups going to be present? It's going to be present on the 1st carbon and it's going to be present on the 3rd carbon. 1st and the 3rd carbon, and nitrogen group will be there. You can open your biomolecules chapter, you will be able to see how they have given it. Okay, now students, so far you understood? All of you understood? Thumbs up in the chat if you all understood. So I told that nitrogenous base is basically divided into purine and pyrimidine, right? It's divided into purine and pyrimidine. Okay, purine and pyrimidine. Yes or no? Yes, all of you? Pyrimidine, no? I put pyramidine. Pyramid. Yeah, so I told that nitrogenous base is divided into purine and pyrimidine, and they're going to have a ring-like structure, heterocyclic structures. Okay, now purines are a double ring structure. Small the name, big the structure. Okay, they are a double ring structure, 9-membered ring. Okay, and on the 1st, 3rd, 7th and the 9th position there is going to be a presence of a nitrogen group. Okay, presence of a nitrogen group. Students, I'm going to write everything on the board. Okay, everything on the board. Your only job is to understand. Okay, only job is to understand and learn after me. Like as I'm telling, you should learn. Okay, and this is going to be how the structure is going to look. A 6-ring structure and then a 6 plus 3. So here one more structure will be there. So here you can see 1st, 3rd, 7th and the 9th position, there is going to be a nitrogen present. Now, if you go to pyramidin, it is going to be a single ring structure around 6-membered ring. Okay, 6-membered ring, and then it's going to have nitrogen in the 1st and the 3rd position. So here you can see 1st and the 3rd position nitrogen is going to be there. Now students, whenever we learn about pyrimidine, whenever we learn about purine, we always talk about who? We talk about adenine, guanine, right? Adenine, guanine, cytosine. So here the way to remember is here you can remember pure as gold. Okay, pure as gold. So I'm going to write it here. So to remember purine, you can remember pure as gold. Pure as gold. P stands for purine, A stands for adenine. Okay, you can remember other ways also, no problem. It's not compulsory that you have to remember this way. And G stands for guanine. G stands for guanine. Yes or no? Clear? So purine, to remember, you have to remember pure as gold. Okay, that is P stands for purine, A stands for adenine, G stands for guanine. These are the people who are coming under purines, adenine and guanine. Now if I have to remember pyrimidine, I'm going to remember with the help of cut. Okay, cut. C stands for cytosine. Okay, C stands for cytosine, U stands for uracil, and T stands for thymine. Okay, T stands for thymine. Okay, got it all of you? We are going to do questions, so now you should know. Exactly, pure as gold. So here what are we going to do? Pure as gold, we are going to cut, okay, with the help of cytosine, uracil and thymine. In this, uracil is going to be present in the RNA, not in the DNA. RNA, not in the DNA. Got it? Pure as gold, cut. Pure as gold, cut. Okay, this is how you're going to remember this. Now ma'am, I understood this. I understood the that is your nitrogenous base. Now we'll go to phosphate. Now in phosphate, there's nothing much to learn. Okay, it's just that P double bond O, that when I'm drawing the bond itself, you will understand. But this is the main part. Purine and pyrimidine is very, very important, and your sugar as well. Okay, and your sugar as well. This is also very, very important because we'll be using these structures to make the bonds. Okay, make the bonds. Clear? Any doubts? Look at this whole thing and ask me if you have any doubts. No doubts? We'll proceed? Okay, let's proceed. So students, as I told you, last one is phosphate. Okay, phosphate, we don't have much to learn about phosphate. Now, how do they form a polynucleotide chain? For that, you should understand that whenever a nitrogenous base, okay, nitrogenous base combines with a sugar, okay, combines with a sugar, I can call this as what? I will call this, okay, I will call this, oh no, yeah, okay, done. Friends, what am I going to call this as? I'm going to call this a nucleoside. Nucleoside. That means we are pushing someone to the side. Whom are we pushing to the side? We are going to push the phosphate to one side, telling that I don't want you. Right? Very good, very good. Okay? Nucleoside. Pushing one person to the side. Okay? Now, to this nucleoside, if I add, okay, let's say nitrogenous base plus sugar plus phosphate, ma'am, you can call this, right, you can call this, what can I call this, students? I'll be able to call this as nucleotide. Yes or no? Yes or no? Yes, very good. I can call this as nucleotide. So whenever nucleoside plus phosphate is going to form your nucleotide, and many nucleotide together will form a polynucleotide chain. Is this clear? Okay, now to the nitrogenous base and sugar, if I add one phosphate, I can call it monophosphate, right? Nucleoside monophosphate, then diphosphate, triphosphate. As I am adding phosphate, I can keep calling it monophosphate, triphosphate, diphosphate, triphosphate. I can keep continuing it. I know that whenever nitrogenous base plus sugar plus phosphate is going to be nucleotide, but I can also call it as, what else can I call it as? I can also call it as nucleoside monophosphate, yes or no? Yes ma'am, you can call it nucleoside monophosphate. Anyone disagrees to this? No, right? Everyone agrees to it? Yes or no? The moment I add one phosphate, I can call it that. Okay. So now students, how do I identify that it is a nucleoside or a nucleotide? How do I identify this? You'll always have this confusion. So always remember, when you have to identify a nucleoside, okay, it's going to be "-ine" in the end. Okay, suffix is going to be "-ine". Okay, at the same time, if it's a nucleotide, okay, if it's a nucleotide, you will identify as it ate. ATE. Sometimes you can also see IC acid. Okay, IC acid. Okay, yes. So 8, you will identify the suffix as 8 if it is a nucleotide, and if it is a nucleoside, the suffix is going to be "-ine". Okay, "-ine". This way you will be able to identify what is the question that they are asking. Suffix is going to be "-ine". Clear? Clear? Line is little cross, or my writing is little cross? Writing is little cross, no? Yes. All of you? Yes, very good, very good. Okay, fine. Okay, fine. Yes. Deoxyribose, everything? Perfect. Perfect. Okay, now we have to form bonds. Okay, now we have to form bonds. Okay ma'am, nucleoside done, nucleotide done. Now how do I bond with the nucleoside, right? I have to make bonds with the nucleoside. So nitrogenous base plus sugar. Let's take a purine or a pyrimidine. Let's take a purine. Okay, this is going to be a purine. First we have to label, students, otherwise we will not know who is going to bind with whom. Okay, 5, 6, 7, 8, 9. Okay, 9 is there. Now I have to bind it with a sugar, right? Let's take a sugar. Okay, done. Sugar is done. I don't think I'll be able to copy paste this. Okay, look here. One sugar is there, one purine is there. Okay, one sugar is there, one purine is there. Now, how will they bond? Always remember, sugar and your nitrogenous base is always going to bind. Now students, if you noticed here, when I was writing there also, I put one dash. Did you notice? All of you noticed that? Ma'am, why did you put here one dash? Here I put 1 dash, 2 dash, 3 dash, 4 dash, 5 dash. Yes or no? That is for a simple reason that purines and pyrimidines, I am going to number 1, 2, 3, 4, 5, 6. Here I am going to number, if I number here also, that is sugar also 1, 2, 3, 4, 5, 6. How will you understand if I am talking about nitrogenous base or I am talking about sugar? To make that difference, to make that clear difference, we have put the dash on the sugar. Clear to all of you? 1 dash, 2 dash, 3 dash, 4 dash. So that now if I tell you that always bonding is going to happen between your 1 dash and 9 position, you will be able to understand. Yes ma'am, 1 dash of the sugar and 9th position of my purines. Right? That difference, to make that difference, we always put the dash. Okay, question can come, keep it in your mind. Now, I told you that the bonding always to form a nucleoside happens between the 1 dash and the 9th position. This is 1 dash and the 9th position. Students, I drew it little ulta. I should have shifted this side. Okay, let's shift it this side. Otherwise, it will be too difficult to draw the bond. Chemistry is testing me. Okay. Okay, done. Now we will label it. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, Yeah, what did I tell? The bond is always going to be formed between the 9th and the 1st position. So this is going to be the bond. Yes or no? This is going to be the bond. Got it? So between my, say, sugar, between my purine or your nitrogenous base and a sugar, one bond is going to be formed. That bond is going to be one, so let's say N, okay, N glycosidic linkage. What is the bond name, students? N glycosidic linkage. How did it form between the purine and the sugar? It formed in the 1-9 position, right? 1-9 position. So when I am forming a nucleoside, always if it is a purine, it is going to bind with the 1st position. Even for your, in your pyrimidine, it is going to be 1 and 1 dash, okay? That means 1st position of your pyrimidine and your sugar. Here it is going to be the 9th position of your purine and the 1st dash of your sugar, okay? So here what is going to form? N glycosidic linkage is going to form. So this is a nucleoside. Yes, ma'am, this is a nucleoside. Now, if I want to make it a nucleotide, how will I make it a nucleotide? Right, I have to make it a nucleotide as well. This is a nucleoside. Yes, this is a nucleoside. But now I want to make it a nucleotide. So let's make it a nucleotide. So what is going to happen? I'm going to remove this H. Okay, CH2OH was there in the glucose, right? That is sorry, in your sugar, right? So I'm going to remove this H and I'm going to add the phosphate group here. Okay, I'm going to add the phosphate group here. That is going to be, students, I'm using a different color. P double bond O, O minus, O minus. This is going to be my phosphate. Can you see? This is going to be my phosphate. Now the entire thing is going to be what? My nucleotide, right? It's going to be my nucleotide. Anyone has a confusion? Anyone has a confusion? Anywhere? This is going to be my nucleotide. Okay, now when I form the nucleotide, one more bond happened here. Yes or no? With a sugar and a phosphate, one more bond happened. What is that bond called? Phosphoester bond. This bond is going to be called as phosphoester bond. Phosphoester bond. Got it? So simple, students, between my sugar and the nitrogenous base, one bond was formed which is called as N-glycosidic linkage. Between the sugar and the phosphate, one more bond was called as phosphoester bond. Okay, phosphoester bond. Clear? All of you clear? No confusion? Awesome. Okay, awesome. So now understood which is a nucleotide, which is a nucleoside. Okay, all of you are clear? Chalo. So this is the bonding. Okay, two bonds you have to study: phosphoester bond and your N-glycosidic linkage. Phosphodiester also is there. That is when I have to combine two nucleotides, right? Many nucleotides will keep adding to form a polynucleotide chain. So when as adding is happening, phosphodiester bonds also will be formed. Okay, phosphodiester bonds also will be formed. As of now, this much is clear. So this is going to be phosphoester. Phosphodiester is when I'm going to combine this with another group of phosphate, another group of nucleotide. That also I'll show you. Now understand, this is ester. One bond, no? Phosphoester. Diester when two are coming. Okay, Asini? Yes, awesome. Clear, ma'am. Clear. Okay. Now, wow. Students, all these notes you will get it on Vedantu NEET English Telegram. Okay, quickly go join the Telegram. Otherwise, you will lose all these notes. Okay. Okay. Now, students, I have to learn how do phosphoester bonds is formed. I have to also know that what is this 5-N, 3-N, right? To make you understand this, I'm going to draw one very complicated diagram, okay, but I'm going to make you understand, okay, but for me, I have to draw it, so it's going to be complicated. So all of you look here, I'm going to draw again a purine, okay, a purine done. Now purine will combine with my sugar, right, sugar. So on the 9th position here, so let's draw sugar. No, I have to draw the sugar to this side. Okay, let's complete the sugar. Here I am writing CH2OH. Okay, yes. Now one bond we are going to draw, that is between first and the 9th position. Am I right or wrong? You should tell me. Okay, first and the 9th position. Now this is a nucleoside. We have to make it a nucleotide. Okay, so that I will, students, can I move this? No, I cannot move this. Okay, 1 second. I'll draw it later more this side. You have to wait until I finish drawing this. Okay, you can also draw with me. You should draw with me so that you will understand what I am trying to explain. Biology teacher drawing chemistry diagrams will obviously take time. Okay, yes. Now here sugar, done. Sugar is there. Here I am going to complete the diagram because here phosphate is going to come and combine, right? CH2O, and here let's draw the phosphate as well. That is going to be P double bond O O O. Okay, done. Now here let's do the bonding. This is an N-glycosidic linkage. Nucleotide done. Okay, nucleotide done. Yes or no? All of you tell me. Yes, ma'am. Pakka. Okay, pakka. Now students, if you all look here, let's say, let's label this. Okay, let's label the ends. Okay, here let's say 1 dash, 2 dash, 3 dash, 4 dash. Okay, this is the 5 dash end. On the 5 dash end, the phosphate is going in binding. Yes or no? So this end is going to be called the 5 dash end. Yeah, 5 dash end. Okay, because why? On the 5 dash end, this was a free end where the phosphate came and bound. Okay, now all of you look here. I'm going to draw an OH here. I'm going to draw an OH here. You'll understand why I drew an OH. Why I did not draw a DNA, but I've drawn a ribose sugar. You will understand now. So students, this is one nucleotide. I told you when two nucleotide combines, only a phosphodiester linkage will happen. So let's draw one more nucleotide. Yes or no? Students, actually, if it was on the other board, I could have copy pasted this, but now I have to manually draw it. Okay. Let's say here we took a purine. Let's take here a pyrimidine. Okay, I'm drawing it here. Let's take a pyrimidine. On the pyrimidine, what's going to happen? On the first position, binding is going to happen. All of you are with me? Yes, ma'am. Okay. On the first position, binding is going to happen. So here I'm drawing a sugar. Okay, here I'm drawing a sugar, and these two are going to bind. Okay, these two are going to bind. This is an N-glycosidic linkage. That is, a pyrimidine is there on the first carbon. This is going to be the first carbon. Okay. Here, this is the first position of your pyrimidine that is going to bind with the 1 dash. Okay. Now let's complete this diagram. Let's complete this diagram, and I will show you what I was trying to say. CH2OH. Okay. Now here CH2OH, I have to combine a phosphate to make it a nucleotide. So let's combine a phosphate. Okay. Let's combine a phosphate. P double bond O O O. Done. Okay, done. Now two nucleotides are there. Yes or no? Yes or no? Two nucleotides are there. This is also a nucleotide. Okay, this is also a nucleotide. Here what is going to happen? The nitrogenous base is going to be a purine. Okay, a purine. This is also a nucleotide. Here the nitrogenous base is a pyrimidine. Okay, pyrimidines. Okay, done. Now I have to combine this both because many like this chains are coming. All of you are writing? Okay, very good. Many of these chains are coming to form a polynucleotide chain. Look here, all of you. I wrote it little too much to the end, but yeah, you people will be able to understand, I believe. Okay, here. Now students, before I make the combination, how do they bind? Here, Here one OH is there, here one OH is there. Okay, ma'am, this we understood. Now from the third carbon, okay, this one third end, this third carbon, this is the third carbon. From this third carbon to this phosphate, there is going to be a bond. What did I say? From the third carbon, there is going to be a bond. All of you are following me on this? Okay, because let's say here there's no OH. Okay, this is our DNA. There's no OH. From here, I'm going to make a bond. Okay, I'm going to make a bond. Let's erase this. I'm erasing this extra carbons. Okay, from the third carbon, there was an OH on the ribose sugar, I mean on the deoxyribose sugar. From there, I made a bond to the phosphate. Okay, agreed. All of you agreed? Yes or no? So on the third carbon, OH was there. Just think of your DNA, I mean your deoxyribose. This is the structure. Okay, here one OH was there. Here H is there in the second carbon. Here one CH2OH is there. Yes or no? On this OH, one phosphate came and bound. Okay, one phosphate came and bound. And with this oxygen, I combined another one nucleotide. Done? Done? So this bond, students, this bond is going to be a phosphodiester bond. Why? Because from here one nucleotide, another nucleotide is binding. This is going to be a phosphodiester bond. If you don't understand this, it's going to be little difficult. So let me know if you are able to understand or not. All of you, are you able to understand or you are finding it difficult? No, right? It's simple, right? Here one O was there, OH was there, that bound here, this formed a phosphodiester bond. This formed a phosphodiester bond. Now here let's again label this. This is going to be the fifth, fourth, third, second, first. Yes. Now students, if I want to make the next binding, if I want to do the next bonding, who is going to be free? 5' end is going to be free or 3' end is going to be free? Because I need an OH to do the binding. Yes or no? I need an OH to do the binding. Which end is going to be free? Which end is going to be free? If you tell the answer for this, that means you have understood the concept. If you're still thinking, that means you haven't understood the concept. See here on the 5' end, one phosphate group has come and bound. Here this was the free end. Okay. Here if I want to make one more bond like this, which end should be free? 3' or 5' end? Here you can see which end is free. Just look and tell me. Very good. Very good. Very good. 3' end is free. Right. So I will call this area, I will call this region as the 3'. Okay. 3' end. I will call that end as the 5' end. 3' end, what am I talking about? That is a 3' OH group is free. Okay. OH group is free. Is this clear? Is this clear? Anyone who did not understand? Again, over and over I am asking the same thing. Let me know. Okay. This is a 3' end. This is a 5' end. That means here we got a free phosphate. Right. Here what is happening? A free phosphate group is there. Okay. Here on the 5' end, a free phosphate is there to do the binding. Free phosphate is there to do the binding. Here on the next end we have a 3' end. So this is the reason why they tell our DNA is parallel, anti-parallel. Why? Because see students, one is going from the 5' end to the 3' end. In the next set, when I am doing the next binding, then 3' end is going to be free, so to the 5' end. Do you understand? Do you understand this? So it is going from the 5' end to the 3' end, 3' end to the 5' end. So in the next one, 3' end is going to be free. For the next one, 3' end is not going to be free. 5' end is going to be free. So this is why they tell it moves from 5' to 3', 3' to 5'. Is this clear? Okay. Now all of you look here. All of you look here and tell me how is the bond formation? How is the bond formation? Now let's label and see. Okay, if you will number here 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, 9. This is I'm also labeling the bonds. Okay, this is an N-glycosidic linkage. Okay, this is an N-glycosidic linkage. Students, this is the only little tricky part. Rest everything is very easy in this chapter. Okay, this is going to be a phosphoester bond. Okay, this is going to be a phosphoester bond, right? Phosphoester bond. Same thing is going to happen here. This is an N-glycosidic linkage. Okay, N-glycosidic linkage. Okay, and this is going to be phosphoester bond. Okay, phosphoester bond. This is going to be a phosphodiester bond. Okay, done. Very nice, very nice. All of you are understanding? Okay. Now students, if you look how the bonds have been formed, you will see that the bonds have been formed from a direction of 3' to 5'. Agree or not? Yes or no? Direction you see 3' to 5'. But how is the nucleotides formed from 5' to 3'? Are you able to understand what I am trying to say? Are you able to understand what I am trying to say? Look here. See, if I am drawing this, see how the bonding has happened. It has gone from the 3' to the 5'. Yes or no? It has gone from the 3' to the 5'. From here to here. Okay, so here the direction of the bond is going to be from 3' to 5'. Yes, direction of bond is going to be from here. See, look here, 3'. Okay, from 3' to 5'. But the nucleotide formation is happening from the 5' to the 3'. That's all. That's all. This is your bond formation. This is the question they will ask. Why? How is this 5' and how is it 3' and this is all about it. Just look, the diagram looks complicated, but if you are listen to me, you will understand this. Any doubts? Just look at the diagram and tell me any doubts. Anywhere you have a doubt, please ask me. Any place you have a doubt. Yes, very good. 3 to 5, okay, 5 to 3. Okay, that is the direction of the formation of your nucleotide. So nucleotide will go from 5' to 3', but bond formation will start from 3' to 5' because there should be a group that is empty here. Okay, there should be a group that is empty here. Done, dusted. Check maadi all of you. Any doubts? Later if you people ask me, where will I answer? Yes, drinking water, students. Where's the water bottle? Yeah. Done. Clear? Pakka? Okay. If you understand this, then everything else is easy. All jujube, small things are really there. Okay, done. Polynucleotide chain. So understood? Many of them will combine to form your polynucleotide chain. Okay, and this is how the DNA strands will be. Okay, DNA strands will be arranged. Okay, done. Now look here, students. Is it visible? Visible, right? So here you can see base is there, adenine is there, ribonucleoside. I told you when side is there, what is the suffix? All of you tell me in the chat box. What will be the suffix if it is side? Yes or no? What will be the suffix if it is a side? Okay, and here it is jaldi ine. Yes, very good. Okay, the tide, it's going to be ine if it is side is ine, right? Tide is ATE. Okay, ATE. Very good. So look here, adenosine, guanosine, cytidine, uridine, thymidine. Okay, ine is there. Here you can see adenosine monophosphate, AMP. You know that ATP, when we break down we get at one point AMP, which you will study in your tRNA when I teach you adapter molecule, but all of you know AMP molecule, right? So after ADP again AMP will come. Okay, so here adenosine monophosphate, guanosine monophosphate, cytidine monophosphate, uridine monophosphate, thymidine monophosphate. So whenever it is side, INE will come. INE will come. Okay, so here you can see adenosine, guanosine, cytidine, uridine, thymidine. Okay, yes. Now deoxyribonucleoside, that means if it is in DNA, it will be deoxyadenosine, deoxyguanosine. Okay, understood? Same thing again here, students. DAMP it will come, deoxyadenosine monophosphate, deoxyadenosine monophosphate. Is this clear? So whenever they give you for identification, you should be in a position to tell if it is a nucleotide or a nucleoside. You know that side means ine. Okay, side means ine, tide means 8. Okay, I am tired of eating. Okay, 8. Yes, 8 also can come, even this can come. Okay, Ic acid also can come, or 8 can come, one among this can come. Okay, most of the time it will be 8 only. Now look here, again I have put that also. Here you can see nucleoside, nucleotide. Okay, so here it is adenosine, here it will be adenylate, adenylate. Okay, tide. Okay, adenylate, deoxyadenosine, deoxyadenylate. Clear? So here they have put the location also where it is present. DNA, RNA, where it is present they have put. Here guanosine, deoxyguanosine, guanylate, deoxyguanylate, cytidine, thymidine, same thing is there, cytidylate. Okay, thymidylate is there. Got it? So this you can just refer. This you can refer because sometimes questions will come. Okay, AMP full form. So that kind of questions you have to know, because again AMP you are going to study again in ribosome. When I am teaching you translation, you are going to study that again. Chalo. Clear, students? Pakka? Pakka pakka? Yes ma'am. Okay. Okay. So this is it. Okay, this is your, one second, yeah. Now we are going to start with salient features of DNA, right? Now we understood the structure of DNA, we understood how the polynucleotide chain is formed, we understood what is nucleotide, nucleoside, all this we understood. Now salient features of DNA. Now first of all, before I tell everything about DNA, nucleic acid, let's learn a little about history. History, students. I finished chemistry, now history. When am I actually going to teach biology? Only God knows. Okay, so now we will go into history, students. So before we tell about DNA, actually there was a person called as Friedrich Mischer. Okay, I'm going to write only Mischer. Okay, Friedrich Mischer. What he did, he was working on some pus cells. Okay, this was again by fluke. That means like, okay, you are going to take some coriander leaf from your fridge, and your mom would have kept coriander leaf in your Amul box, ice cream box. You will think it's ice cream, but you will see inside coriander, right? Like that. By fluke, you got coriander. Same way, Mischer was working on something, and it happened that there were some pus cells. Okay, pus cells, and that was taken. Okay, someone was doing something, and by fluke, they saw pus cells, and this Friedrich Mischer noticed that in this pus cell there is something living. Okay, something is there because it was present. Okay, that living thing was present inside the nucleus. He called it as nuclein. He called it what? Nuclein. So this was the first discovery ever. Okay, he was working on something else. Some people tell he was working on some pus cells. Some people tell he was working on owl. So multiple knowledges are there about this. Okay, so our parents have a job of customization. Whatever box you give them, biscuit box, ice cream box, yogurt box, any box, they'll fill it with their own things. We will think that, oh, my parents out of the blue they bought ice cream and kept in the fridge, and you will be so excited, exhausted, open the fridge and you see that inside that what will be there? Yesterday some curry will be there, or some tomatoes will be there, onion will be there, anything can be there. Okay, anything can be there. So that is how it is. Okay, that is the reality of Indian parents and their connection with ice cream boxes. Okay, that's why they make us buy also boxes. Otherwise, they'll be like, okay, you go get that family pack and come. No, they'll be like, buy boxes, so that they get the box. And by mistake, you give box to your friend, and your friend does not give box, students. Your mother's hate list, first that fellow's name will be there. Forget about people who cheated her, forget about people who ran with her money, no tension. That box, how dare you give the box and not bring it back? Tell me, students, you people answer. This is first, that blacklist book will be there, no? Top your friend's name will be there. So that is the relation, okay? So Misher worked on pus cells and he found nuclein. Then one more scientist came. Obviously, scientist, as if scientist is less. That fellow called as Altman. Okay, Altman. He told, wait. This nuclein has something very acidic. Okay, this is very acidic. He told, this is acidic in nature. So I don't want it to be nuclein. I will call it nucleic acid. I will call it nucleic acid. So understood how the name came? Nucleic acid. This was the discovery. Okay, this was the discovery. True, no? See, I know. Okay, so in not P cells, pus cells, pus cells. How will P cells, pus cells? Okay, Misher worked on pus cells and they found nuclein. That nuclein, then Altman came and he was like, wait. There's something acidic about this, so I do not want it to be called nuclein. I will call it nucleic acid. Okay, I'll call it nucleic acid because it has something acidic. This was the first discovery. Now let's go into the salient features of DNA. Students, before I go into the salient features of DNA, let's draw one beautiful DNA. Okay. Too thin it became. It looks like one hair braided now. Are you all enjoying the chapter? Tell me that. Are you all enjoying the chapter or is it boring? Is it boring or enjoying? Okay. Tell me, how was the chapter so far? Come on, students, let's make it 100 likes, all of you. Hey, what is this, da? Okay, so look here. I drew a DNA, right? I drew a two polynucleotide chain which are wounding around each other. Okay, wounding around each other. Now, when I see this, no, students, this orange color, imagine this is the backbone. So, the backbone of DNA, okay, backbone of DNA is made up of whom? It is made up of sugar and phosphate group. Sugar plus phosphate, not your nucleoside, okay? It is sugar plus phosphate. So the first ever salient feature of your DNA is that it is made up of two polynucleotide chain, okay? Polynucleotide chain. Got it? The first thing is it is made up of two polynucleotide chain which are wound around each other, and its backbone, okay, its backbone is made up of none other than sugar and phosphate. Now you will be thinking, hey, you were teaching so much about nitrogenous base. Now where did nitrogenous base go? Nitrogenous base is going to project inside. Okay, they are going to project inside, students. They are not acting as a backbone, but they are going to be the ones who are going to keep these people in place. Okay, they are the ones who is going to keep these people in place. Got it? So they are going to project inside. Got it? Project inside. So this nitrogenous base, write NB, okay, is going to project inwards, okay, to make sure that these people stay in place, right? Because these both are like this. To keep them in place, what do we need? We need someone to project inwards so that they are together. That is the first salient feature of your DNA. Now if you look at the DNA, it is a double helical structure. Now for that also there is one story. Okay, double helical structure was actually found. Okay, this double helical structure was actually found with the help of an X-ray diffraction. Okay, double helical structure was actually found by 4 scientists, we can tell. Okay, who are the 4 scientists? All of you know. Tell me, who are the 4 scientists? Let's start with Rosalind Franklin. Okay, Rosalind Franklin, who Pavam did not get Nobel Peace Prize. Okay, then we have Watson. Crick and Wilkins. Okay. Now what did they do? What did they do? Very good, very good. Who is that? Rani. Yes, awesome. Okay, Madhumati, very nice. So these are the scientists, Rosalind, Watson, Crick and your Wilkins. These 4 people worked on understanding the DNA structure. So they did an X-ray diffraction. In the X-ray diffraction, they understood that okay, the structure of DNA is actually a double helical structure, not chumma like that, okay. It is a double helical structure. That is how the name called as double helical structure came. But students, only these 3 people got the recognition for that. Rosalind Franklin did not get recognition, because usually when a person, you know, is not alive during that whole process, they will not be able to award, and also it was like it was not recognized, okay. So these 3 people got recognition for finding the double helical model, but Rosalind Franklin did not get the recognition. Clear? Okay, done. So this is the second most characteristic. Now this double helical structure, that means the name double helical structure comes. Now if I look at the strand, I can see, as I already mentioned, this is going to be from 5' to 3'. Imagine, yeah, 3'. This is going to be from 3' to 5'. Yes. So always remember that both of your strands are always going to be anti-parallel. Both of your strands are always going to be anti-parallel to each other. That is the third salient feature, that the strands are, okay, strands are anti-parallel, okay, anti-parallel. Got it? That is, one is going to be 3' to 5', another one is going to be 5' to 3'. Now you know the significance of it, yes or no? Do all of you know the reason of it? Yeah, Nobel Peace Prize is only for the people who are living. Like if they are dead, it's not going to happen. Okay, so students, strands are anti-parallel. So 3 points we understood in your salient features. 2 polynucleotide chain that forms the double helical structure. The backbone is made up of sugar and phosphate, and the nitrogenous base project inwards. You can see the structure here. This is your nitrogenous base, okay, NB. Okay, they are projecting inwards. Now, double helical structure, it was found by Rosalind Franklin, Watson, Crick and Wilkins with the help of an X-ray diffraction. Okay, this they will ask you, okay, with the help of X-ray diffraction. Got it? With X-ray diffraction, and the strands are always anti-parallel, 3' to 5' and 5' to 3'. Now, the next points we will do. Yes, we will do PYQs, Jay. We will do some of the PYQs, not all of the PYQs, some we will do, some we will keep it for the later, okay. Chalo, is this clear, all of you? Now, I actually want this diagram again, but again I have to draw. Okay, let's do one thing, students. Let's duplicate this. How do I duplicate, yeah? Okay, I duplicated. Now, I don't know what I'm doing. Okay, wait all of you. Okay, done. No duplication is happening, so I have to take another slide. Yeah, students, done. Yes, we are going to go to the Chargaff rule. Don't take tension. Okay, now students, anti-parallel, parallel, clear, all of you? Okay, now always remember the two chains, the two strands are going to be moving in a right-handed fashion. Okay, it's always going to move in a right-handed fashion. Right-handed fashion. What is the pitch of the DNA, students? What is the pitch of the DNA? Thank you. What is the pitch of the DNA? All of you tell me. Pitch you have studied by now. 3.4, 34 Armstrong. What is it? What is this? The pitch of the DNA or the pitch of the helix, right? The pitch of the helix is going to be 3.4 Armstrong. Okay, 3.4 Armstrong. That means they will do a 360 turn. Okay, a complete 360 degree turn will be done. Now, understand when such a turning is happening, okay, when such a turning is happening, 3.4 nanometer, no? Oh yeah, 3.4 nanometer or 34 Armstrong. 3.4 nanometer. Okay, 3.4 nanometer. Now students, when they are turning, right, when they are turning, now this is your DNA structure, when they are turning, in one turn, how many base pairs will be there? How many base pairs will be there? Let's think like this. How many base pairs will be there in one turn? 10 base pairs will be there. Yes or no? All of you know this or not? Okay, in one turn, okay, in one turn there will be around 10 base pairs. One turn there is going to be around 10 base pairs. Done? Now we have to calculate the distance between, okay, we have to calculate the distance between two base pairs. Distance between two base pairs, can we calculate or no? Yes ma'am, you can calculate. How is it? It is easy. Pitch divided by the number of base pairs in one turn. How much is it going to be? 3.4 divided by 10 I can do. Yes or no? 3.4 divided by 10 I can do. That is going to be 0.34 nanometer. 0.34 nanometer. So this is how I'll calculate the distance between two base pairs. I hope this is clear to all of you. Okay, I hope this is clear to all of you. Now in a DNA, in a diploid, right, not in a haploid one, in a diploid one, say it is around 6.6 into 10 to the power 9. Yes, all of you agree? 3.3 into 10 to the power you would have seen. Now in diploid, how much it is going to be? 6.6 into 10 to the power 9 into, I am putting this, okay, let's convert this into, it's going to be 3.4 into 10 to the power minus 9. Okay, 10 to the power minus 9, that is going to be 3.4 into 10 to the power minus 9. Okay, so I am going to calculate the length of the DNA. Okay, I am going to calculate the length of the DNA by the total into the distance between the two base pairs. Okay, I am going to cancel out this and this, and I am going to do 6.6 into 0.34. Instead of that, one more way to do is I can do 1 divided by 3.1 something, okay, because 1 divided by 3 is going to be 0.3333. You can't round it off, so it is around, so here it is going to be 2.2. So the length of DNA is going to be around 2.2 meters. Okay, this is going to be the length of DNA. You can directly learn it, but if you want to do maths like me, then you can do this. Okay, done. So students, it is going to move in a right-handed fashion, and we have a B form DNA. Okay, we have multiple forms, A, B, C, D. Okay, B form DNA, Z. This is going to be a B form DNA which you will have to study. Okay, the most common types, and the pitch of the helix is going to be 3.4 nanometer. In one turn there will be around 10 base pairs. Distance between two base pairs is going to be 0.34 nanometer, and the length of the DNA is going to be approximately 2.2 meters. Very good, all of you have answered. Awesome. Okay, those who joined right now, did you all mark your attendance? Did you all mark your attendance? I don't think so. I can see that you haven't. Okay, you haven't. Mark it quickly. Okay, chalo students. Now we will learn some numbers. Okay, we have to learn some numbers. What are these numbers? See, some organisms have given, and they have given the length of the DNA. Now we calculated the length of DNA. Here they have given haploid content. We calculated with the diploid content, right? We have to calculate with diploid content. So here they have given bacteriophage 5174. It has around 5386 nucleotides. Okay, 5386 nucleotides. Bacteriophage lambda has around 48502 base pairs. Students, I tried maximum to find a trick. Then I saw multiple other people doing tricks like this. Okay, that is 4 plus 1 is equal to 5, 4 minus 1 is equal to 3, 7 plus 1 is equal to 8, 7 minus 1 is equal to 6. But I found it very complicated, so I'm not going to teach that way. Okay, but if you feel it easy, you can definitely check it out. Okay, students, just because I don't like the subject maths, whichever chapter I'm taking nowadays has maths. whichever chapter. Breathing and exchange, maths. Today I was doing, today I have to do excretion in DNA batch. There also osmolarity, some amount of maths. Why is there so much maths? Okay, so look here students, bacteriophage 5174 is around 5386. Bacteriophage lambda is around 48,502 base pairs. E. coli is 4.6 into 10 to the power 6 base pair. Okay, 10 to the power 6 base pair. Then haploid content of DNA is going to be 3.3 into 10 to the power 9. But you know, if it is diploid, it's going to be 6.6. Okay, now if you look here, you will see one very nice thing. What is that? Here they have told nucleotide. Here they have told nucleotide. That means this is a single stranded structure. Okay, everywhere else you can see this is a base pair. That means this is all double stranded structure. But this first one is going to be a single stranded structure. They will ask you a question asking which among this is single stranded, which among this is double stranded. So you should be in a position to say that bacteriophage 5174 is single stranded because here it is given nucleotides. Here they have given nucleotides. They have not given base pair. That means base pairing is not happening. Done? Clear? Yes or no? This you have to by heart. Okay, this you have to by heart. Yes. One second. Yes, yes. This is from Tatva book. The table is from Tatva book. I told you, no, nowadays I am referring Tatva books. You will get boot camp. You will get boot camp for the topics that you have missed. Sujana Shri, yes. Done? Let's move. Basically, okay. Now students, Chargaff rule. This we did not see, no? We did not see. So in salient features, one of the features is that no matter whatever happens, okay, no matter whatever happens, one thing is going to be constant. That is, purines is always going to be equal to the pyrimidines. Purines is always going to be equal to the pyrimidines. This is always going to be constant, no matter what. Now let's say I draw one. Okay, I draw one. That is, say AUGUACCT. I don't know what I'm writing, but yeah, TCG. Okay, I've written one strand. This is one strand. Okay, let's say it 5 dash to 3 dash. Now all of you tell me, the strand that we are going to draw down is going to be 3 dash to 5 dash. Okay, so yes, students, always remember that purines and pyrimidines are going to bind with each other. Okay, purines and pyrimidines are going to bind with each other in such a way. We know that purines, okay, we know that purines have two people. Who is that? Pure as gold. A and G is there. Okay, pyrimidine also has two people. You throw that uracil for now that side. Okay, now pyrimidine also has two people. Pyrimidine also has two people. Who are the two people? So how do I remember who is binding with whom? The easiest example is that right now you people are in my class. So that is going to be the example. At Gopika Ma'am, you tell in your mind, okay, respect should be there. At Gopika Ma'am's class. So at Gopika's class, that is A will always bind with T, G will always bind with C. Okay, this is how you will remember at Gopika's class. So students, A will always bind with T with 2 hydrogen bonds and G will always bind with C with 3 hydrogen bonds. Is this clear? Is this clear? Got it? So no matter whatever changes happen, okay, at Gopika Ma'am's class, perfect. Okay, at Gopika Ma'am's class. Yes, okay. One second. So here adenine, thymine, guanine, cytosine. So now you know this. With that you tell me this. A will always bind with who? A will always bind with T. U. Why did I write U, students? T. Okay, wait. A, T, G, U. Okay. T will always bind with A. G will always bind with C. How many times I have written U? This is DNA strand. No U. Okay. G, A, T, C, O, Y, U. C, A, T, cat. A, G, C. Okay. Then G, A. Okay, something I've written. Now look here. A, T, A, C, A, T, T. Okay. A, G, T, A, C, G, T, T. Okay, done. Check madi, all of you. Correct? I've done 2 strands. Now let's chuma see the count. Numbers. Okay. Now, how many A's are there, students? 1, 2, 3, 4, 5, 6. 6 A's are there. That many T also should be there. Otherwise, I'm wrong. Okay. 1, 2, 3, 4, 5, 6. Perfect. How many A's are there? That many T's are there. So, A double bond T is pakka ratio in this. Okay. Now, next one. Let's take next one. Okay. Next one. T. Okay. T. 1, 2, 3. 3 T's are there. That many A's should be there. 1, 2, 3. Perfect. Okay. So, this is standing both the ways right. A to T, T to A. Done. Now, G. Let's count G. 1, 2, 2 G's are there. Okay. That many C should be there. 1, 2. Perfect. How many G's are there? That many C is also there. So, this proves that no matter what, purine is always going to be equal to pyrimidine. Is this clear? Now, one more thing is that this rule was found by Erwin Chargaff. Okay. Erwin Chargaff. So, this rule was called as Chargaff rule, where he told that the ratio of purine should always be equal to the ratio of pyrimidines. Okay. And this rule was called as the Chargaff rule. Chargaff rule. Now, one thing students get a lot confused is that, ma'am, you told this. Okay, I told this. Yes, I did tell that A is equal to T and C is equal to G. But did I ever say that this two should be in ratio? I never told that. How many ever AT is there, let them be there. How many ever GC is there, let them also be there. I never told that, see, AT, okay, A is equal to T should be equal. Okay, same way C is equal to G should be equal. But I never told these two people should be equal. I never told that. So, these two people need not be equal. That means there can be regions which are rich in AT. Okay, there can be regions which are rich in AT. No problem. There can also be regions which are rich in, you know, this also, what is that? CG. No problem. If this is rich, maybe this will be less. This will be less. Maybe this will be more. Or maybe this will be less. This will be more. No issue. These two doesn't have to be equal. These two doesn't have to be equal. Students make mistake in this. Students think that, okay, because this was equal, even this should be equal. Not at all. Not at all. Okay? Not at all. This rule is only when purine should be equal to pyrimidine. It's not like AT and GC should be rich. No. Same. No, no. Okay? Done. Got it? Okay. Awesome. Okay? This is Chargaff rule. Okay? Always remember, ratio of purine should always be equal to ratio of pyrimidines. Done. Next, what are we going to do? Packaging of DNA. Students, how long is the DNA? Tell me when you want a break, okay? When you're tired, tell me. I'll give you a break. I'll give you a break. Okay, now tell me, how long is the DNA? Okay, how long is the DNA? Length of your DNA. All of you. Quickly, tell length of your DNA. 2.2 meters, right? Imagine, students, this 2.2 meters. Let's say my size of my eukaryotic cell. Which color is this? Yeah, size of eukaryotic cell will be around 10 to 20, okay, 10 to 20 micrometers. Okay, this is how much the size of your eukaryotic cell will be. But my DNA is around 2.2 meters long. My DNA is around 2.2 meters long. Now, how do you expect that in a small nucleus, right, in a small nucleus, this entire 2.2 meter of DNA to fit in? Not possible. Yes or no? It's not possible. So, we came up with a solution. We told, okay, let's do one thing, let's do packaging. All of you have seen this, people making, your grandparents would have made sometimes. You would have seen making this chain, okay, small, we kids used to make, right? We used to take the bead, we used to put the thread, put the bead, put the bead, put the bead, put the bead. At the last, we tie one knot so that this bead does not move that side, this side. And then we used to make that white color pearl chain. All of you have noticed. Same way, you would have also seen your grandparents doing that flower, right? For the head also we keep, for God also we keep. So for those flowers also you would have seen, they'll keep many flowers, tie a knot. Many flowers, tie a knot. That means that entire thread we are making into a entire floral, I'm out of words, okay, out of words, the chain, okay, the chain. Same way, I am going to take this 2.2 meter long, it's like one thread only, 2.2 meter long thread into, I don't know why I'm running out of words. I'm going to push, I'm going to coil it and keep it inside the nucleus, okay. I'm going to coil it and keep it inside the nucleus, okay. So, my DNA is around 2.2, approximately 2.2 meter long. Now, for this, I need help. I am going to do packaging of DNA. I'm going to do what, students? Packaging of DNA. With the help of who? I am going to take some positively charged proteins. Okay, I am going to take some positively charged proteins called as who? Histones. I am going to take some positively charged proteins called histones to do the packaging. Clear or not? Okay, ma'am, histones, okay, histones is going to come and they are going to do the packaging. But how many histones is going to come? Who is going to come? All of this you want to know? Yes, definitely I want to know. So, there are around 4 people now who is going to come, okay. One more person will come, that is mass entry, okay, mass entry. So, 4 people are going to come. Who are these 4 people? These 4 people are going to be, first is going to be H1 protein, no, H2A protein, okay, H2A, H1 I told of. That is actually suspense, students. H2B, okay. H3, look at my dedication, one color I am using. H4. These are the 4 histone proteins that is going to come. That means my DNA is negatively charged. Negative will attract positive, opposite poles attract each other. So, the negatively charged DNA will be wound around, that is, this DNA will be wound around a positively charged histone proteins. Simple problem, 2.2 meter DNA, I will be able to keep it inside my nucleus. Agreed, all of you? So, which are the histone proteins that are going to help? H2A, H2B, H3, H4. These are the varieties, okay? Now, they will not be like, I will come single, will come double, they'll tell. So, let's make them double. Okay, let's draw the structure. Look how they're going to be. 1 H2, okay. Then students, H2B, okay, H2B, then H3, H4, H4, okay, H4. They're going to be like this, okay. Now, I told you they will not come single, so they will have one person also with them, that is copies, okay. They'll have two of them. See, I could have put one diagram and explained you, but I want to draw and make you understand, okay. So, look here, this is how the structure is going to be, okay. Yes or no? They all look similar, but this is the structure. So, students, this structure, understand, this structure is your proteins, H1 proteins, H2, H2A, H2B and all that, okay. Now, these structures, as I told you, they will not come alone. They will have two of them, okay. They'll have two of them. So, here your next one, okay. Here your next one. Yes, and last your green, okay, green. Okay, now let's label them. Who are they, students? H2A, okay, H2A, H2B, H3, H4. So this is how they came. Okay, ma'am, understood. Okay, they came as an octamer. That means octamer is already formed. Yes or no? So I will call this as histone octamer. All of you agree or disagree to this? You have to agree, students. What you will do by disagreeing? Okay, you have to agree that this people came as a histone octamer. Histone octamer. That means very simple. Very simple. These people came 2 each. Okay. Done. Okay, 2 each they came. They formed an histone octamer. Now what am I going to do with histone octamer? Okay, very nice. So what am I going to do with this histone octamer? Now I have to make sure that this DNA is going to wound around this histone octamer, and it is going to condense and form my chromosomes, and then I can keep it inside the nucleus. This is my task. Yes, ma'am, this is your task. Okay, everything is my task only now. So this is histone octamer. Now we are going to draw how it is actually going to look. Okay, how it's actually going to look. Now let's say, students, this is your DNA. Okay, this is your DNA. This is your DNA. Okay, long thread-like structure. Now we have your histone octamers. Okay, now we have your histone octamers. 1 histone octamer, 2 histone octamer, 3 histone octamer. Okay, done. Now we have to wind this DNA against it. Yes or no? Okay, we have to wound this histone octamer. This is how it is going to bind. Okay, this is how it is going to bind. Now after that, what is going to happen? They'll keep condensing. Okay, now they'll keep condensing. And that condensation will look like this. Okay, octamer, 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 octamer. Okay, condensation is happening. They're going to go zigzag, 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 zigzag. Okay, zigzag, zigzag. I'm showing you condensation. Okay, you should be very proud. Okay, then after this, they will keep condensing again and again. Okay, they'll keep condensing again and again, like this. Okay, condensation. Yes, you understood how chromosomes will be formed? Okay. Don't tell like this nonsense. What is Saturn and all you people are telling? Okay, this is my DNA. Whatever I draw, you have to agree, students. So what is going to happen? Let's see. This is your DNA. Okay, this is your DNA, which is around approximately 2.2 meters. Okay, fine, ma'am, done. Okay, now this DNA wound around an histone protein. Okay, this is a histone octamer, and the DNA was wound around. So whenever the DNA winds around the histone octamer, I will together call this entire complex as a nucleosome. Got it? Students, this notes you will get. Okay? Yes, this notes. Thank you. Students, this notes you will get on Vedantu NEET English Telegram and as well as my Telegram, Biology by Gopika. Okay? Don't miss out on this notes. I'm drawing all this only for you people. Okay? What will I do by drawing all this, students? So look here. This is how the binding happened. This is to give you a 3D understanding of how this is going to happen. So DNA was there. I wound it around a histone octamer. When histone octamer plus DNA together, it will be called as nucleosome. Okay? Together it will be called as nucleosome. Understood? So this nucleosome, it will have around 200 base pairs. Okay, 200 base pairs will be there, and winding will keep continuing, students. Winding will keep continuing. It will get more condensed here. Can you see how condensed it is getting here? More condensed here, and finally after lot of condensation, it is finally going to form my chromosomes. Okay, it is finally going to form my chromosomes. Yes or no? This chromosomes you would have studied in your botany now. Yes, this will now get into my nucleus. Okay, this will now be able to enter into my beautiful kutti nucleus. What is the nucleus color? Yeah, Okay, it will be able to enter into my nucleus. Are you able to understand now? Are you able to imagine how it got into the nucleus? Yes? Yes, yes, we'll take a break, okay? This got into the nucleus. Okay, this got into the nucleus. Done? Okay, done. So this is how my entire formation happened. Now, which are the main proteins? Okay, which are the proteins that is rich in this entire histone and your nucleosome is your lysine and arginine. Okay, lysine and arginine. Okay, these are the two amino acids, lysine and arginine. Okay, it will be arginine, correct students, spelling, just check the spelling. Okay, just check the spelling. Lysine and arginine. Chumma chumma, don't tell pakka pakka. Tell correctly. Are you able to understand or not? Okay, are you able to understand or not? G9, correct, no? Okay. Now students, I forgot about one person. Who is that one person? Students, now in this nucleosome complex, now say I put your DNA. Okay, now I'll put this dark pink color DNA here. It's going to go like this. It's going to come like this. Wow, wow, wow. This is not how art works. Wait. Okay, like this, like this. Like this, it's going to come out. Okay, to keep this in place, to keep this DNA in place, I have to have one protein. Yes or no? That protein is going to be called as my linker protein or your H1 protein. Because as I told you, when you are tying the bead also, you put a knot. Why? So that the bead does not move that side, this side, right? So I will use the help of a H1 protein that is also called as a linker proteins, which will keep everything in place. Okay, linker proteins that will keep everything in place. So where this is going to come here, it's going to come here. Okay, it's going to come here, going to come here, and going to come here. Okay, got it? Linker proteins will do the rest of the job of keeping this people in place. Linker protein. Done? Now students, at lower level, okay, histone proteins are more than enough, no doubt in that. But when higher level we need to make the entire chromatin, we will use another set of proteins which is called as non-histone chromosomal protein. What will we use? We will use non-histone chromosomal protein for higher, okay, winding. Okay, this is in higher chromatin fibers. We will use NHC, which is non-histone chromosomal protein. Okay, chromosomal protein that will actually help us for what? This is in for higher binding. Okay, higher coiling, we can tell, higher coiling. Okay, this entire thing is coiling, condensation is happening. Okay, condensation, condensation, condensation. Finally, this will be able to enter into our nucleus. Is this clear? Yes or no? So the packaging of chromatin at higher levels will require us an additional set of protein which is called the NHC. So this is going to be our additional set of protein. Okay, this is going to be our additional set of proteins. Okay, additional set of proteins. Is this clear? Done, done, done? Okay, additional set of proteins. Anything did we miss? Anything did we miss? Or are we good to go? Done, right? Done, right? So, very simple, nucleosome, don't forget, histone plus your DNA forms your nucleosome. Around 200 base pairs will be there, around 1.75 rounds they will go, okay? That is again not needed for you. But here they have asked problems about it, 200 base pairs, okay? Then students, you have H1 protein, which is the linker protein, and lysine and arginine is the maximum. Now they will condense together so that they can fit into the nucleus, okay? They can fit into the nucleus. Then we take the help of NHC, which is another additional set of proteins that will actually help us for chromatin fiber formation in the higher levels. Done, Done? Okay. Now one more thing in this topic is, students, when I look under the microscope, no, when I look at these people under the microscope, when I'm looking at these people under the microscope, they will look like a bead on the string structure. What will they look like? Bead on the string structure. It's very simple. See, look here. I'm putting some dots. Okay, dots, 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 dots. Okay. Now if I connect it with a thread, I'll connect it with a thread. Na, na, na. Okay, connected with a thread. It looks like a bead on a string structure. Yes or no? It looks like a bead on a string structure. So under the microscope, this one is called as a bead on a string structure. Don't forget this word, bead on a string. Bead on the string structure is how it will look under the microscope. Done and dusted. Everything is covered. Okay, everything is covered. Yes, ma'am, everything is covered. Okay, very good. Now we have euchromatin and heterochromatin. Okay, euchromatin and heterochromatin. Where did that go? Yo, students, I put that, students. Where did it go? Okay, I'll write it here, central dogma. Okay, euchromatin, heterochromatin and central dogma, will divide it into two parts. Okay, okay, give me a 2 minutes break. Okay, give me a 2 minutes break. That is, resume back. What is this? Okay. Resume back at 11:45 AM. No, sorry. I'm in PM still. My brain is in PM. Okay, 11:45. 11:45, 46. It's already 43. Students, why am I like this? Students, 11:50. Okay, maths too difficult to do maths. Let's do 11:50 AM. Okay, I'll be back. Yes, don't go anywhere. If you go anywhere, then it's there for you people. Okay, yes, you people also have something and come back. Yeah, students is not coming out. Okay, see you all.
recording now.
Yeah. Hi guys, I'm back. I'm sorry, little long break it happened.
Drinking tea.
Yeah. Wait, I have to tell them. They went off. This is why I don't give them break. One break, they'll run away. Join back. Yes. Okay, students, ready for the next topic? Hello. See, DNA will continue 22nd if batch is launching, it will continue from there. But you don't have to worry because you will have boot camps. Boot camps are basically where we will take a complete marathon of what has been done, and you will also get the video recordings of that. Okay, so don't take tension about that, and your test also will be according to that. Okay. Okay, volume is high. Okay, done. Ready. Euchromatin, heterochromatin we have to do. After that, some experiments. Then what are we left with? 1 second, students. This book. Ready. Not audible. I'm audible. Very audible. Okay. Chalo. Now let's start central dogma. Before we start central dogma, we have to learn something called as euchromatin and heterochromatin. Now what is, ma'am, euchromatin, heterochromatin? Now say I draw a chromosome. Okay, say I draw a chromosome. Okay, let's do in this side of the board, students. This is your centromere. Okay, I draw one chromosome like this, and I draw one chromosome like this. Okay, this is how I draw a chromosome. Done? Okay, it looks little outside. That's at the side, but yeah. Wait, we have to fix the diagrams. Okay, one chromosome like this, one chromosome. Okay, that means now in this chromosome, say I am drawing areas. Okay, few areas I am drawing. Some condensed areas I am drawing, some free, loosely packed areas I am drawing. Now, what is this, ma'am? What are you drawing? I am not understanding. So, look here, students. The portion, okay, the portion where it is dark, right, it's condensed, okay, that portion is called as heterochromatin region. So in the same chromatin, okay, in the same chromatin there will be two regions, one heterochromatin, one euchromatin. Hetero means darkly stained or tightly packed, okay, darkly stained or tightly packed. So this region I will say heterochromatin, yes or no? So I'll put this region, okay, this will be my heterochromatin. That means, why did I say it was heterochromatin? Because they are densely packed, right? They are densely packed. And because they are densely packed, if I put a stain, will they get bright or will they be dull? They will definitely be dark, right? So they will be darkly stained, okay? Darkly stained. Yes or no? Understood? So heterochromatin is the region where the DNA is condensed. Okay, I can say that the DNA here is very condensed. Agree? All of you agreed? Okay, DNA is very condensed. Very good. Now, this is the region. Now, students, something called as transcriptionally active, transcriptionally inactive words are there. That is very simple. The one, see, what is transcriptionally active or inactive? From your DNA, we have to form mRNA. From mRNA, we have to form protein. Yes or no? For this DNA to mRNA formation, we have to use some enzymes. For that, if that enzyme will be able to be placed or not. See, here the enzyme will be placed. Yes or no? Here the enzymes will be placed. The space is there for the enzyme to be placed. But in the heterochromatin area, there is no place for the enzyme to be placed. So I would say this is transcriptionally inactive. This region is transcriptionally inactive because it does not provide the area or the space for that enzyme to come and sit. Okay, so this area becomes transcriptionally inactive. Now let's go to this area. Okay, this area. euchromatin. This is your euchromatin. This is going to be opposite of this, exact opposite of it. So what will it be? First thing, it is loosely packed. It is loosely packed. Second thing, I have put darkly stained, so this will be lightly stained. Lightly stained. Yes or no? Lightly stained. Then I told DNA is const, here DNA is little loosely packed. Okay, DNA is here loosely packed. Then I told transcriptionally inactive, but here it is going to be transcriptionally active because it gives the space for the enzyme to come and sit. Okay, for the RNA polymerase to come and sit. So this portion, is this clear, students? Is this differentiation clear? Yes or no? This differentiation is clear? Okay, clear. All of you are telling me clear when I am hoping that it is clear. Why is it so thick? Yes. Ajiju. Done? Okay, now we have to learn central dogma. The entire molecular biology revolves around the central dogma. Now your understanding of this chapter entirely depends on if I am making you understand or not, and if you are going and reading NCERT or not. Yes or no? These two factors are the main thing that will affect. Same way, your entire molecular biology is based on the central dogma. That means the purpose of you studying this chapter is because of these steps. What are these steps? Simple things, students. DNA to mRNA, mRNA to protein. This is your central dogma. Now wait, ma'am, only this you have written. What about other things? Yes, what are the other things? When DNA becomes mRNA, I will call it as transcription. I will call it as what? Transcription. That is, DNA becoming mRNA will be called as transcription. When mRNA becomes protein, I will call it translation. I will call it translation. Okay, done. Now, when DNA can form its own copies, from DNA, if it can form its own copy, it will become replication. That means it can form its own replica. Replication. Agreed, all of you? Now, there are cases where mRNA can become DNA. Who can tell the answer for that? When mRNA become DNA, what will I call it? That is present in your HIV virus. HIV virus is the problem. Who is that? HIV is the problem. HIV will do all this kinds of thing. Very good. Okay. So that is when mRNA goes to, that is RNA to DNA. Okay, RNA to DNA, I will call it reverse transcription. I will call it reverse transcription. Reverse transcription, and this is because of the enzyme reverse transcriptase. Reverse transcriptase. Yes or no? Done? Okay. This is your central dogma. That means these are the ruling factors. Okay, that DNA should become mRNA, mRNA should become protein. Protein is the one who is going to decide the traits which we have to come from the paternal, maternal. How do you look so much like your mother, father? All this resemblance is because of your protein able to code. Right, protein able to code and synthesize. So DNA should become mRNA, mRNA should become protein. DNA to mRNA is transcription, mRNA to protein is translation. From RNA to DNA is called as reverse transcription, and that happens because of the enzyme called as reverse transcriptase. Done, 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 done. Now we are going to our first ever experiment. Are you all excited? So far, how did you find the class? Did you understand everything? Is there anything that you have a problem with? See, we have done so much. Right, we have done so much. Everything I have written. Okay, everything I have written. See, bonds, types, everything. Okay, everything is here. all you have to do is go back and read your NCERT. Yes or no? Go back and read your NCERT. Now we are going to do the experiments. Okay, done? Chalo. Now students, the first ever experiment. Now we kept telling ma'am DNA is the genetic material, DNA is the genetic material. Okay, fine, DNA is the genetic material. But who proved it? How did the first finding happen? Yes, we understood that nucleic acid name came from your Altman and your Friedrich Mishra. No problem in that. We also got to know that DNA is a double helical structure. But how did they know that DNA has all the information of a cell? How did they know that DNA can inherit something? Chapter name is what? Molecular basis of inheritance. How did they know that, okay, something can be inherited from the maternal and paternal? All this is a, like, it's like a history, right? It's a mystery. So we are going to decode that mystery. The mystery starts from Griffith. Okay, so Griffith was working on Streptococcus pneumoniae. What was he working on? Streptococcus pneumoniae. Okay, he was working on this. While he was working on this, he noticed that there are two strains, okay, two strains of Streptococcus pneumoniae. What is it, students? Two strains, okay. The first strain was called as the S strain or the smooth strain, okay, S strain or the smooth strain, and one was called the rough strain or the R strain. Rough strain or the R strain. Understood? Now, ma'am, why are you calling it rough, smooth? How did you understand all this? Very simple, students. Rough strain, okay, rough strain had a polysaccharide or a mucopolysaccharide coating. Okay, this is your rough strain, I mean a smooth strain. On that, there was a coating. Okay, there was a coating. A clear coating was there. Okay, yes or no? This coating was there. Now, this coating indicated that, okay, this is a smooth strain. Okay, this is a smooth strain. Fine. This was a polysaccharide coating. Polysaccharide coating. Did rough strain have any of this? No, rough strain is parvum. It did not have any of this. Okay, it did not have any coating, nothing. It was parvum just like that. Okay, so because of this polysaccharide coating, some people started saying that, okay, this smooth strain is actually virulent. That means it can cause disease. Okay, it can cause disease. And that is because, see, normally students, something is there, something is not there. This is acting little different, this is acting little different. It is normal common sense to think that it might be because of the polysaccharide, right? So they started saying that this S strain is virulent or it can be cause disease. Okay, it is disease causing. Rough strain is not virulent. Okay, rough strain is non-virulent. This was the finding. Okay, virulent. Okay, now what did Griffith do? He was working on mice. What was he working on? He was working on mice. And he was injecting these strains, okay? S strain, R strain, all this he was injecting. And he was like, okay, let's see what is going to happen. Okay, now let's see what happened actually. Now here if you look, you will notice that first he gave live S cells. That means S strain was given. The mice obviously died because we already know that S strain is going to be virulent. No doubt in that. Then he gave then he gave R strain. R strain let the mice will obviously leave, no, because R strain is non-virulent. Then he did one thing, okay, let me do one thing. I will heat kill this mice. Not the mice, students. I will heat kill this strain. Okay, he told, I will heat kill this S strain so that if there is something that is actually killing this mice, let it not be there. Okay, so he heat killed the S strain, and for his expectation, the mice lived. That is, no cells from the healthy mouse. That means nothing happened to the mouse lived. Then he was like, okay, let's do one thing. See, in the result, this one, let's say this is A, this is B, this is C. From this, where and all the mice lived? In these two case situations, mice live, that is B and C. So he mixed B and C. Now you tell me, if you were Griffith, if imagine you're Griffith, Rani is Griffith, Pavitra is Griffith, Madhumita is Griffith, all of you people are Griffith. What would you expect? You are mixing B and C. Both the situation the mice lived. What would you expect? You would expect that the mice would obviously leave, no? Yes or no? If you were Griffith, you would have expected this. Even Griffith expected this. But for his shock, what happened? When he mixed B and C, that is heat-killed S strain and living R strain, he noticed that there is some living object that has got transformed. That has got transformed, okay? Justice for mice, it seems. Here for NEET students only, justice is not happening. Then you will get justice for mice. Huh? Here from past 3 hearings have happened. This should go as a roast video, students. Okay. Yes. So here Griffith noticed that during this entire experiment, he noticed that something has happened. What was that something? He noticed that some living object has got transformed. Okay. Okay, I'll say some living content. Okay, some living content has got transformed. Okay, has got transformed from whom to whom? Transformed from the heat killed, very important, okay? Heat killed S strain to R strain. And he did not know the name. He was like, I don't know the name. I was not planning to do this experiment. This just happened. So I will simply call it as the transforming principle. What did he call it as? The transforming principle. So actually this experiment is the principle of all the experiments. Hershey and Chase, Avery, McLeod, McCarthy, all this one's principle is going to be transforming principle, because only because of this experiment they understood that okay, maybe there is something that we have to work on. Yes or no? So heat killed S strains, from heat killed S strain, something got transformed to the R strain that made, okay, that made R strain virulent. Okay, that made R strain virulent. And he called it what? He called it the transforming principle. Easy experiment, very easy experiment, yes or no? Expected result did not come, but another result came, and with that result other scientists also came. Got it? Other scientists was like, okay, wait, I can do something about this. I can do something about this. Any doubts? Let me know. So far, any doubts? Yeah, something. Hello, honey bunny. Okay, here honey bunny is not there, mouse is there. Okay, yes. Okay, done. Ma'am, Griffith's experiment we understood. Now you tell me about Avery, McLeod and McCarthy. So students, obviously, scientists is not less. So next scientist came. Okay. Okay. Next scientist came and they were like, okay, now that you have told that this is transforming principle, we are kind of sure that it is DNA, because what else could get transformed? But they did an experiment. What was the experiment? The experiment was very simple. Let's take one shape. Okay, so the experiment was 1, 2, I think I can duplicate this, no? How do I do that? Let me not do things I don't know. Okay, 3. Ayyo. 1 second, students. Me and this board has some issues. We're not that good with each other. Okay, 2 and 3. Okay, yes. So this 3 things, this 3 as the test tube. Okay, think of this 3 as the test tube. So in each test tube, one thing was there. Okay, one protein was there. Okay, one protein was there. Okay, one protein was there. one RNA was there, one DNA was there. Okay, these three were there. Again, same thing I'm going to continue, okay? Till that you have to wait. One protein, one DNA, one RNA. There's an easy way to do this. That is one color I could have taken and done it, but you won't understand. Okay. Students, in a test tube, they took all the three things. What are the three things that they have doubt? It should be neither a protein, it should be neither an RNA, or it should be a DNA. These are the three things that you have doubt. You will never doubt carbohydrate, lipids. You will not doubt. You will know that, okay, maybe it should be a protein, because in the entire central dogma, we are talking about these three people, DNA, RNA and protein. So it should be a protein, or it should be a DNA, or it should be an RNA. Okay, fine. Avery, MacLeod and McCarthy did this experiment, okay? They did this experiment. First, what did they do? They took protease. Okay, what did they do? They took a protease. Okay, this protease is a thing that can digest the protein. Okay, they took a protease. That means that can digest the protein. Yes, exactly. They took enzymes which are made up of protein like protease, RNAs and DNAs, and they started digesting these people. So, because this protein got digested, right, this protein went from here. Okay, protein went from here. When the protein went, what did they expect? They thought, okay, if protein is the genetic material, then pakka transformation wouldn't have happened. But unfortunately, transformation happened. Transformation happened. Yes or no? Transformation happened. Obviously, because DNA is still active, no? So, still transforming principle is going to happen. Then, okay, fine. Next thing they did. Next thing, what did they do? They took RNAs. Okay. Protease went. Now, they took RNAs. Okay, they took RNAs. They took RNAs and they were like, okay, let's see if this is actually the villain. Okay, if this is actually the villain or not. But students, what happened? Here also transformation happened. Okay, here also transformation happened. Now, if I see it in the case of mice, like if I keep here mice and see what will happen here, the mice will die here also. The mice dies. Okay, because again, transforming principle. Here also the mice will die. Yes or no? The mice dies. Okay, so here 2 out. Okay, last one, what did they take? Obviously, last one they took DNAs. Okay, last one they took DNAs. When they took DNAs, protein went, protease went, RNA went, DNAs went. Okay, now they took last one. Here they noticed that there was no transformation. They noticed that there was no transformation. That means what, students? The mice lived. Okay, the mice is alive. Are you clear with this? Are you clear with this experiment? Yes or no? So first they took protease. Protease digests the protein. Okay, transformation happened because DNA is still there. Second time they took RNAs. RNAs digested RNA, but again transformation happened because again DNA is there. Then they took DNAs which will digest DNA. They noticed that no transformation had happened and the mice lived. So now it was easy for them to tell. They were like, okay, DNA is the genetic material. Okay, DNA is the genetic material. You accept or not, DNA is the genetic material. They told after this, okay, that hence proved DNA is the genetic material. Genetic material. But problem happened. See, everyone was talking about transformation, but many scientists disapproved to the whole thing. They were like, there's no clarity. There's no clarity that, okay, DNA is the genetic material. So this experiment completely was disapproved. Okay, this experiment did not see the light. That is basically biochemical reaction. That means checking the biochemical nature of the transforming principle is the experiment name. This did not work because transformation was not like, people are not able to understand how from one cell to another cell something will get transferred. They were not able to understand how using protein you will be able to tell that protein is not the genetic material, because if you see, okay, protease is there, RNAs is there, DNAs is there. All three are proteins. Using protein, you are telling that protein is not the genetic material. So some contradictory statements came. Then they told, okay, this experiment is completely scrapped out. Like, it's not going to help us at all. Is this clear to students? Is this clear? Any doubts? Okay, I've tried to explain as much as possible with a simple diagram. Yes? Yes, ma'am, clear. Haha. So many people who are crying for the mice. No one is crying for me because I'm sitting and writing all this. No sympathy. For all the mice, monkey, donkey, they'll have sympathy. For humans, no sympathy. Yes? So, here, done. This experiment got disqualified, okay? Disqualified happened, students. Now, the next experiment, which is the most widely accepted experiment, is your Hershey and Chase experiment. These people were like, wait, we will make some sense for you and show, okay? We will make some sense for you and show that why DNA is the genetic material, okay? 1 second, student, leg is paining. Yeah. Why is DNA the genetic material? That we will try to show with the help of Hershey and Chase experiment. What did these people do? Hershey and Chase experiment started with taking a bacteriophage. Okay, they took a bacteriophage. They took a bacteriophage. This is your bacteriophage. Inside the bacteriophage, there is your genetic material, okay? DNA is there. Okay, DNA is there. This is what they took. Okay, they took the genetic material. So, the outside coat is your protein coat. Okay, outside coat is your protein coat. Coat, okay? And inside one is your genetic material. Now, why is this given? To attack the cell. Okay, to attack the cell. Bacteriophage means what? Virus that will attack a bacteria. So, this is your bacteria, this is your bacteriophage, it will attack. It will attack means what, students? It will attack the E. coli and immediately release the genetic material inside. This is what is going to happen. Okay, this is what is going to happen. So, they took two things. Okay, they took two things. These two things you have to keep in mind, all of you. Okay, did I miss something? Students, something is little missing. Wait, okay, no, it's not missing. Okay, I've put. Yeah, students, look here. So, they took simple thing. What is there in the protein code that is not there in the DNA? What is there in the DNA that is not there in the protein code? This is the difference that they have done. Okay, difference that they have done. Here, in protein, understand there are some sulfur proteins. So, they took sulfur. Okay, they took sulfur, that is, they took sulfur 35. Sulfur 35, which is radioactive sulfur. Here in this one, DNA was there. In DNA, we all know phosphorus is there, but in protein that is not there. So, they took P32, radioactive. Now, it is understood that if DNA is the genetic material, we will see radioactive phosphor in the supernatant or pellet, depending on centrifugation, right? Okay, this was the thought. Okay, now, if protein is the genetic material, we will see sulfur 35. This was the clear-cut differentiation. Now, no one will be able to question, oh, how did this come? They will not be able to question because it will be clearly seen in the pellet and it will be seen in the supernatant. Now, what did they do? The first step that they took was infection. That means taking the bacteriophage, infecting with the E. coli cell. Okay, infecting with the E. coli cell. So, they took that. Now, all of you tell me, first question to all of you, E. coli cell, how many base pairs do they have? We just studied the box. Let me see how many of them remember. E. coli cell, how many base pairs they have? Yes, this Hershey is very funny, okay? Those who are joining new, no, those people are not sitting in the class, not there smashing the like button. No attendance, nothing. Always bunky. Tell me all of you, how much is it? No searching, no, don't open your textbook. Don't open your textbook. Tell me E. coli, I'll give you clue. 4. Very good, Rani. 4.6 into 10 to the power 6. Right, that is your E. coli. This is how you have to connect and study. Now, students, first job was infection. That means here they took one, here they took one, S35 they took, P32 they took. Both of them got infected. Now, we all know DNA is the genetic material, that is transforming principle. Now, what happened? Once they infected, both of their genetic material got in. But simply just looking here, you will understand this is not radioactive. Here, this is radioactive. Okay, after infection, they did blending. This is also called as Blender's experiment, okay, because we are using blender, too many blenders. Why we are using blender? To remove the protein coat, whatever is not required, no, to remove from the E. coli cell, because the protein coat will be attached like this. Once they leave the genetic material, no use of this virus. We all know virus is mainly to leave the genetic material, rest everything genetic material will do. They will catch like this, no? Blending say this will come out. Blending say this will protein coat will come out. So they removed the protein coat. After that they did centrifugation. Now students, during centrifugation we'll get two things, a pillet and a supernatant. What will we get? We'll get a pillet and a supernatant. Okay, during centrifugation the heavier particle, okay, the heavier particle will go to the pillet, that is the one that will settle, and the lighter particles will be in the supernatant. Lighter particles will be in the supernatant. Now what happened after? What is the result that we got? We noticed that there is no radioactivity detected in the cell. No radioactivity. If protein was the genetic material, then it should have got transformed, yes or no? But no radioactivity in the cell. But where did we see all the radioactivity? In the supernatant. So we noticed that radioactivity of S35 detected in the supernatant. You know, genetic material usually will be little heavy. They will settle in the bottom as pillet. And also we will notice that radioactivity is there in the cell because this has got transformed, it has got inside, yes or no? So now in P32 what happened? Radioactivity detected in the cell and no radioactivity detected in the supernatant. So this was very clear that okay, supernatant is basically the liquid form. The lighter particles will go up and the heavier particles will go down. So the lighter particle will settle in the supernatant and whatever is heavier will go to the pillet. Imagine I mix mud in this. In this water I mix mud. Mud will settle down. Whatever liquid part will be there, I can call that the supernatant. But for that I have to do centrifugation. I have to go around, okay. Some RPM I have to rotate, and after that what is going to happen? On the top some liquid will be there, on the bottom some liquid will be there, okay. Pillet and supernatant. Now students, is this clear? Very easy experiment, but this proved the transforming principle, yes or no? This proved the transforming principle. What was proven? Very simple. In S35, let's write S35, okay, the radioactivity. In this radioactivity, where was radioactivity seen, students? In this case, radioactivity was not seen in the cell, okay. Radioactivity was not seen in the cells, okay, not seen in the cells. Where was all the radioactivity? Radioactivity was seen in the supernatant, was seen in the supernatant. Done, done. So by this what did we prove? Protein is not the genetic material. Chalo. Now the next radioactivity, that is your P32, okay, radioactivity. Students, you should know why did we take sulfur and phosphorus. You have to know, because this is only present in DNA, this is only seen in protein. Mixing up will not happen, okay, mixing up will not happen, okay. So P32, what happened here? Here they noticed that radioactivity, okay, radioactivity is seen in the cell, in the E. coli cell, okay, in the cell, and no radioactivity in the supernatant. No radioactivity in the supernatant, okay, in the supernatant. Clear? Okay. So from this experiment, what did we conclude? From this experiment we concluded that DNA is the genetic material. DNA is the genetic material and protein is not, okay, protein is not. Clear? Any doubts? Any doubts? Okay, done. Okay, done. Now properties of genetic material. Now we spoke about the experiment. Which are the three experiments we went through? Griffith's experiment that explained us the transforming principle. Griffith did not know how this got transformed from the S strain to the R strain, but he called it a transforming principle. After that, Avery, MacLeod and McCarthy came up with the biochemical nature of this transforming principle. They tried to prove that DNA is the genetic material. They kind of proved that DNA is the genetic material, but it was not widely accepted by other botanists and the zoologists. So they disapproved it. After that, Hershey and Chase came with a bacteriophage experiment where a virus that infects bacteria, where they took bacteriophage, E. coli cells, they took radioactive S35, radioactive P32 and they did an experiment. In that, first step was infection, second step was blending, third step was centrifugation. In the infection, the bacteriophage infected the E. coli cell. In the blending, they removed the protein coat, the genetic material got inside. They noticed that after centrifugation, in the protein, that is your S35, they noticed that no radioactivity is found in the cell. That means nothing got transformed. Okay, all the radioactivity was found in the supernatant. Okay, now in the other side at P32, they noticed that all the radioactivity is in the cell and there is no radioactivity in the supernatant. By this, Hershey and Chase experiment proved that DNA is the genetic material and DNA is the transforming principle that transformed from the S strain to the R strain and made the R strain virulent. Got it? So this is the conclusion of all the experiments. Now we have to understand what is the criteria for being the genetic material. See, if DNA is chosen as the genetic material, why is that so? Why is RNA not taken as a genetic material even though it was the first ever genetic or the first ever nucleic acid? So this we have to understand. Okay, this we have to understand. Done? Okay, there are 4 points to, like, students, now for election, if the Prime Minister, right, now we have Narendra Modi has to stand, there are some criterias. Okay, so that criterias, if he go through, then only he'll be able to stand for the election. Winning, not winning is a different story, but to stand in an election, you have to have some criteria. Can anyone go and stand? Tomorrow I want to stand. Oh, I'll go and stand. I want to become the Prime Minister. Desperately, I want to become the Prime Minister. I cannot go and stand, okay? There are some criterias for to go and stand. So, same way, DNA and RNA also has some criteria to be the genetic material. Let's see what are the criteria. The first ever criteria is that they should be able to replicate itself. Okay, they should be able to replicate itself. Replicate itself. Okay. Now, next thing is they should be chemically and structurally stable. Chemically and structurally stable. Okay. What is happening here? What is this? How can we start the DNA batch from starting dear? Students, DNA batch, don't worry. Yeah, I don't understand what is it. Yeah, understand whatever is done in the DNA batch, we are doing marathons on YouTube. Okay, marathons on YouTube. So you can watch the marathons on YouTube, join the DNA batch, continue your classes. Now see, body fluids and circulation is done in DNA. I have finished body fluids and circulation. Same way, I forgot the chapter name only. Body fluids and circulation done. Structural organization in animals, I'm going to take. Okay, I'm going to bake. I'm going to bake, it seems. I'm going to take. Okay. Same way, Shreyas sir has finished units and dimension. He has done a marathon here. Same way, Diksha ma'am has done. Baswa sir has done biological classification. Baswa sir has finished. I finished it here. Okay. Got it. So, we are doing it simultaneously on YouTube and your DNA batch. So, whatever we have finished in DNA batch, you can check it on YouTube. And after that, you join the DNA batch and continue in the flow. How will we start from starting, students? I think June 1 batch joined. Okay, when June 1 batch joined, no, May 18th 1 batch joined. How will we tell them to we'll start from starting? Okay. Exactly. GM for RNA. Okay, students, so chemically and structurally stable, they should be able to undergo slow mutation. Okay, mutation should happen, but that mutation should be slow. Okay, it's not like today you sleep as a man and tomorrow you wake up as a dolphin. That much fast mutation should not become, no? So that one. Okay. Chalo. Then the last one is they should be able to express themselves. not like cry and tell that, oh, I'm so sad. Okay, they should express itself in Mendelian characters. Okay, Mendelian characters. Got it? Now, DNA and RNA. Now, we'll see who has won the race. Okay, students, we have to see who is actually winning it. You already know who is winning it, but still, as a teacher, I have to teach. This is also one important question they have asked many times. Okay, chalo, we'll put tally for both of them, who is going to win and who is going to lose. Who are you? Are you team DNA or RNA? All of you? Are you team DNA or RNA? Wow, students, this is like my batch DNA and my batch RNA. Oh no. Very late my brain works. Okay, my brain is only content, molecular basis of inheritance. Other things and none of it is working. Okay, students, tell me, are you with team DNA or RNA? All my RNA students of YouTube should be with RNA. Okay, Papa students, RNA is the first genetic material, nucleic acid. Okay, yeah, these people all took off team DNA. Only Hasini is RNA. Okay, students, you are RNA batch, your RNA YouTube series. Why are you taking DNA? Huh? We are all team. Okay, even I am with you people. We are team RNA. Okay, if DNA people see this video, they are going to comment this long message. Okay, they'll be like, ma'am, you forgot us, that this. Okay, very nice. Okay, so students, this is team DNA and RNA. Now, first question is, can they replicate themselves? Okay, can they replicate themselves? Yes, both of them can replicate themselves. So, both of them are getting a point. Okay, both of them are able to replicate themselves, both of them are getting a point. Second point is chemically and structurally stable. Here one problem is that we saw that deoxyribose sugar and ribose sugar. In that, who was there? In deoxyribose sugar, one oxygen was less. In ribose sugar, one oxygen was more. Because of this, gutbud happened. Because of this, reactivity and stability changed for whom? RNA. So here RNA gets no points, DNA gets all the points, because in DNA multiple factors are there, not just that, multiple factors, students. First is that absence of oxy group. Okay, absence of that oxy group. Second thing is thymine in the place of uracil. Okay, thymine replaced with uracil. No, uracil replaced with thymine. What is this? Okay, uracil replaced with thymine. Okay, these are the main two reasons why this DNA is chemically and structurally so stable. Okay, done. Now the next one is mutation. Yes, both of them mutate, but the problem is, okay, both of them mutate, but the main problem is DNA mutates slowly, but RNA mutates fast. See students, in DNA, this class, we will take it slowly, right? One portion, 1.5 hours of each. So, we will divide it into multiple pieces and we will take this chapter. But on YouTube, I will take a 3 hours class and finish half the portion. So, in my RNA batch, I am little faster. In my DNA batch, I am little slow. So, same way, mutation is slow in DNA and fast in RNA. We do not want fast mutation. Like I told you, today if you sleep as Rani, tomorrow I don't want you to wake up like elephant, or I don't want you to wake up like giraffe. Okay, anyone like that. Okay, anything. So, that much fast mutation should not happen. So, that you should be Rani tomorrow also, right? So, yes, that's the same thing. So, here it is slow, here it is fast. Okay. Now, express itself as Mendelian characters, that is to form the central dogma, that is from DNA to mRNA to protein, right? To express itself, they should be able to go through the whole thing, that is from DNA to mRNA. Students, basically they should be able to form proteins. Okay, they should basically be able to form proteins. Now, if you look here, in 3 steps, that is 2 steps, DNA to mRNA, mRNA to protein. In 2 steps, they will form proteins. Here in 1 step, it will form protein, because only this much is there, no? RNA to protein. So, both of them are doing this job also well. Okay, both of them are doing this. Tick, tick, but this is happening in 2 steps, this is happening in 1 step. So who is the winner of the challenge? Who is the winner? Who has all the ticks? Who is the person who can be the genetic material, or who is absolutely the best one to be the genetic material? All of you tell me in the chats. DNA is going to be the genetic material. Okay, so DNA has that because many things are exactly right when we compare to the properties of the genetic material. So DNA is widely accepted as the genetic material for majority of the organisms. Done? Done. This is your properties of your genetic material. Differences also we did. Differences also we did. Now RNA world. So difference is this only, okay? This box itself is the difference. Okay, and you should know these things also. Absence of an oxygen group made it more chemically stable. Uracil replaced with thymine also made them chemically stable. Got it? So this is the difference. I'm not writing it again. Now let's go to RNA world. Students, tell me when it's 1 o'clock. Okay, tell me when it's 1 o'clock. I have another class. Okay? Yes. Now RNA world. My students are RNA series students, my precious batch of YouTube students. Now, my RNA students are not any less, or not like that. RNA is not any less than your DNA. RNA is not any less than your, yes, exactly. Presence of that extra oxygen in the structure makes it more reactive, okay, in RNA, okay. Now, my RNA students and DNA students are a no comparison. Both are amazing in their own way. Same way, RNA, just because RNA is not the genetic material for majority of the organism, that does not mean RNA is completely useless. RNA does some of the things that even DNA cannot do. Like what? RNA is actually, students, you will not believe, it is the first genetic material. I mean, you will believe. I don't know why did I say that. Okay, RNA is the first genetic material found on earth. Okay, it is the first genetic material that was found on earth. Yes. Okay, so this RNA does so many things that DNA cannot do. I told, no, what are the so many things that they cannot do? Let's see what are those so many things, students. Okay, students, this RNA mainly is of 3 types. Okay, RNA is mainly of 3 types. 1, 2, 3. Okay, first one is RNA will form mRNA. Types of RNA, first one is mRNA. M stands for messenger RNA. Anyone has a doubt in this? M stands for messenger RNA. Okay, messenger RNA. As the name suggests, what does messenger do? Okay, what does messenger do? Thank you so much, Ashwati. Okay, what does messenger do, students? It is mainly going to send messages, send signals, right? Send messages, send signals. So this messenger RNA is going to communicate between your DNA and protein, telling them what you have to do. Because on the messenger RNA, there is going to be genetic code. On the messenger RNA, who is going to be there? Genetic code will be there. This genetic code has to be read and the protein has to be continuously synthesized. Yes or no? So messenger RNA will do the job of, if messenger RNA is not there, protein formation will not happen. So messenger RNA is very, very important. Okay. Okay. Then the next one is mRNA. Then we have tRNA. What RNA? tRNA. Okay. What RNA? tRNA. Or we can also call it sRNA, that is soluble RNA. Okay. sRNA, that is soluble RNA. What is this, students? Very simple. tRNA, sRNA. tRNA or your transfer RNA acts like a, okay, acts like a adapter molecule. Transfer RNA acts as a adapter molecule. Adapter molecule. That means what? Now, what is an adapter's job? Now here there is no adapter, but all of you would have seen adapters in your home, right? So it will be kept and we will put multiple plugins to it. Now for normally one switch, I can only put one plug, but instead I'll put an adapter and that adapter is going to do the job of what? One, it is mainly going to connect to the main switch, and with that I am also going to connect other connections to it. So same way, your tRNA or your adapted molecule, what it's going to do, from one on the top there will be amino acid acceptor, so it will accept the amino acid. At the bottom it is going to read what is on the mRNA. Oh, AUG is there, methionine is there, okay, UUU is there, phenylalanine is there. So it will read, attach, read, attach. Finally, it will form the protein synthesis. So adapter molecule is exactly doing the job playing between two people, doing two jobs. One on the top where it is accepting the amino acid, one in the bottom where it is going to read what is written on the mRNA. Got it, all of you? So tRNA is a transfer RNA. It acts like an adapter molecule. Okay. Can DNA do this? No, DNA will not be able to do this. Then the last one is, students, rRNA. Okay, last one is rRNA, which is also called as ribosomal RNA. Okay, it's called as ribosomal RNA. Definitely this is going to help in the formation of ribosome. Also, it is also going to help as a catalyst. Okay, it is going to have a catalytic action. Okay, it is going to act as a catalyst. It's also going to help in the formation of ribosome. Okay, it's also going to help in the formation of ribosome. Here ribozyme, right? Ribozyme, the enzyme. Okay, it's going to have catalytic action, enzymatic action. Okay, enzymatic action, and it is also going to form ribosome. Enzymatic action. It's called a ribosome. Done? Done? Clear? So RNA is no small fellow, he is big fellow. Okay, he is doing so many jobs, right? He is doing so many jobs. So he's not just doing one thing, he's doing multiple things, and then multiple things are very, very precious things. Okay, that has to happen in your central dogma. That has to happen in your central dogma. Done, students? Any doubts? Anyone noting it down? I will wait for you to note it down. By then I'll do decoration. Students, these pretty notes are made just for you. So download it and take it from the Telegram, okay? Don't wait for me to tell it. Exactly. RNA batch reading about RNA world. Yes. Done? Okay. Okay. Writing, huh? Okay. So I hope this is clear. So RNA is not any small person. He is doing so many jobs, right? Even in the central dogma, mRNA is going to do that entire job. Okay. Done. Okay. Now the next thing. Now students, replication. I think mostly we will stop the class here until replication because I have one more class. So I will not get lunch break. Okay, I have a batch after this. So we will stop up to replication. I'll try to teach you how much I can do about replication. From enzyme action, we will take it in the next class. Okay, tomorrow's class I'll let you know, students, because I have a I have to visit some students, so I might go for that. So I will let you know. Okay, I'll let you know when the class will be taken. Okay, okay. Now replication. Students, replication means what? In central dogma, we already understood that DNA and your RNA can replicate themselves, right? Replicate itself. That means make copies. Yes or no? Agreed, all of you? Make copies of themselves. So this replication also is the same thing. Make their own copies. But in DNA, there is a thing. 3 ways is there to replicate. Okay, 3 ways is there to replicate. Now your DNA, you already know it replicates in semi-conservative manner, but understand. If you understand this, just understand the colors, okay? So students, let's say parents are going to be purple color. Okay, these are your parents. Each of the parents will help as a template. Each of the parents. Now imagine I am giving you one paper. Okay, say this is the paper I am giving you. This is one paper which I have written. I have written this paper. Fine. In this paper, this is the original copy. I am giving you this copy and I am telling you take a Xerox. So this becomes the template. The one that you are going to take Xerox, the one that you took Xerox is going to become your daughter DNA or the new template, right? This is the actual one which I gave you. This becomes the template. This will become the copy that you took or the daughter DNA that you took, okay? So the parent DNA is going to be purple color. Each of this parent DNA is going to act, okay? Each of this parent DNA is going to act like a template. So 1, parent DNA, one daughter DNA will form. Okay, one daughter DNA. Fine. This is one way of doing it. Okay, again one parent DNA, one daughter DNA. Copies have been formed. Now after this, basically what is going to happen? Obviously parents are going to go back to each other. Yes or no? Parents will go back. Okay, parents will go back. And the daughters also will go back to each other. Okay, daughters will also go back to each other. This condition, this kind of a replication is called as conservative replication. What is this called as? Conservative. Okay, conservative. Now this is what happens normally. Replication can happen with conservative, disruptive and semi-conservative. Here it is conservative. Now what is happening in our DNA? Students, all of you would have noticed, I'll tell you a very simple story for this. You and your sibling, how many of you have siblings? Just raise your hands in the chat. You and your sibling will be fighting. Your parents will take, let's say dad will take elder daughter's side, mom will take younger daughter's side, or if it's son, depending on that, they will take one side. Okay, so let's say your parents were together. Okay, your one parent took one of your side, another parent took another child's side. Okay, after fight, normally parents will go back together and the daughters or the siblings will go back together. But in some households, okay, in some cases, okay, some very vichitra cases, what is going to happen, students? Very simple thing will happen. Parent will come for support. Okay, imagine we both are fighting. Okay, we both are fighting. Parent took my side. Okay, took my side. Okay, fine. We are sorting out the issue. Again, my dad took my sister's side, imagine. Okay, we are sorting out the issues. But after the fight almost got over, what is going to happen? They both will start fighting, we both will start fighting. They came to solve the fight. But in between, some dialogue came that they also started fighting. Now what is going to happen? They are not going back to each other. So they will continue, right? We are going to continue as a pair. Pair matlab what? We both will become like a team. Okay, we both will become like a team. And we'll tell nonsense about them. They will tell nonsense about us. So this is how most of the houses it happens. You tell me if it happens in your house. Okay, if it happens in your house. Yes? Okay, so usually the parents have to go back to each other, the daughters have to go back to each other. But sometimes when the fight goes out of hand, when they forget whose topic it was and they start telling their flaws and then it just gets so, so bad, then they both will still continue as a pair. This two will still continue as a pair and they will keep talking about each other. We'll go, my dad is like this, my mom is like this. This will keep happening. So this is how your DNA replicates. This is the easiest way to explain it. Semi-conservative manner. Okay, semi-conservative manner. Semi-conservative manner means what, students? One parent, one daughter. One parent, one daughter. Or one template, another template. One heavy, one light. Anything can happen. Okay, anything can happen. One heavy can be there, one light can be there. Or one parent will be there, one daughter will be there. Is this clear to all of you? So I'm going to label this, okay? This is your parent DNA, okay? P1, P2, again P, D1, okay? Daughter 1, P2, daughter 2. Don't think I don't like son and all, because I cannot write it here. Daughter DNA is what is mentioned in your NCERT. Okay, for my marzi, I cannot write whatever I want. Okay, this is your daughter DNA. Same thing goes here, parent, daughter, parent, daughter, parent, daughter, parent, daughter. So semi-conservative manner is shown by your DNA. Okay, so DNA replication happens in a semi-conservative manner. Is this clear? Is this clear? All of you? Okay, all of you face this, huh? Not bad, yeah. I thought only I face it. Not bad. All of you face it. Okay, it looks like a national problem now. Okay, it looks like a national problem. Now I have stopped solving fight, because when I solve fight, no, it becomes worst. It's better that I keep quiet, because whenever I solve fight, it becomes worst. It last becomes on my head only. Exactly, Nikita. Students, so we should stop solving fights. Okay, it's not our cup of tea. Okay, we cannot be advocates, because at last our issues will come out. What we did 10 years back will also come out. Why you want to embarrass yourself? So whenever you are seeing a fight, take a popcorn and eat the popcorn. Okay, for that also you'll get scolding, but whatever. At least it's better than listening to your own things, what you did 10 years back. 10 years back you did this. Exactly. Okay, done? Okay. Students, I have put PYQs, yeah? What are people questioning me? I've put PYQs, but I don't know if you'll be. Chalo, we'll do PYQs, huh? How much time I have? I'll explain this one experiment and then we will stop. Okay, machinery is no time. One experiment I'll explain and I'll make you understand. Okay, see, look here. Look here, students. Meselson and Stahl were the people who found out that DNA does semi-conservative replication. That means one parent DNA, one daughter DNA, one parent DNA, one daughter DNA. This you will never forget in your life with this example, okay? And those who haven't smashed the like button, please do smash the like button right now. Mark your attendance, because once you mark your attendance, I feel like, okay, you were there in the class, okay? And you're enjoying the class. So Meselson and Stahl were the people who did the experiment where they took two things, okay? To prove this semi-conservative manner, they took an N15. Okay, this is not a radioactive isotope. Okay, N15, that is a heavy nitrogen. Okay, they took a heavy nitrogen. That is, they did not take just N15, they took NH4Cl. Okay, NH4Cl with N15, heavy nitrogen isotope. Okay, then they took N14, that is light nitrogen. Okay, light nitrogen. This is an isotope, and this was also taken as N14HCl. Okay, now they let the E. coli grow in this. Students, why did they take the E. coli? All of you remember the answer for this? They took the E. coli because E. coli replicates, okay, E. coli replicates in 18 minutes to 20 minutes. Okay, duplication happens in 18 minutes, replication happens in 20 minutes. So they took E. coli. E. coli is easier, no? Because replication should happen. If imagine something's replication happens after 48 hours, how can you wait? So that will not happen. So what did they do, students? They let the E. coli, okay, first they let the E. coli grow in this NH15 medium. Okay, NH15 medium. 15 medium. After they grew the E. coli in NH15 medium, obviously both the strands will be NH15. Any doubts in this? Both will be N15, N15. Agreed? They let the E. coli grow, both will become N15. From N15, they dipped it into N14 medium. Okay, they dipped it into N14 medium. I don't know what I'm doing. Wait, students. Yes, they put the same thing into N14 medium. They're letting it replicate, okay? This went into N14 medium. So what is expected? What is expected here? They got hybrid, okay? They got one N15, one N14, okay? Same thing, again they got one N15, one N14. Got it? They got hybrid here, okay? They got hybrid here. Okay, they got N14, N15, N15, N14. Yes or no, students? So this all happened in your E. coli. Okay, E. coli was taken. Why E. coli was taken? Because it replicates, right? Its DNA replicates in 20 minutes. Replicates in just 20 minutes. So they wanted that. Okay, so first 20 minutes they dipped it here. Next 20 minutes, they dipped it here. Did you understand? Did you understand, all of you? Yes or no? Now look at this diagram, you will understand better. So first N15, they dipped it in N15. Okay, centrifugation was done. After 20 minutes, they dipped this same thing into N14. Okay, first they dipped it in the parent DNA N15. After that, they dipped it in N14. When they dipped it in N14, they noticed that they got hybrid. Okay, they got hybrid. That means, okay, they got hybrid. That is, here you can see N15, N14, N15, N14, hybrid they got. Again, they dipped it for another 20 minutes in your N14. Okay, again they dipped it in N14. So what they would have got, 40 minutes, they got N14, N14, N15, that is 2 hybrids. So they got 50% hybrid, okay, they got 50% hybrid and they got 50% light, 50% light. In next round what they will get, students? They will get, see, in the next round, so this is in the first case, okay, in the first case, very simple, they got 50% hybrid, 50% light, okay. Again I keep it for 20 minutes, okay, now again I keep it for 20 minutes, then what will I get? I will get 75% hybrid and I will get 25% light. Agreed or disagreed, all of you? You have to understand this, problems will be asked, okay. I got 75% hybrid, no sorry, light, and I will get 25% hybrid. Because why, students? Because the N15 will be fading, right? The N15 will not be there more because we are not dipping it for a longer time, okay, we are not dipping it for a longer time. So N15 will keep fading, so I will get 75% light, 25% hybrid. Again if I keep, I'll get 85% light and I'll get 12.5% types hybrid. So this will keep continuing, 20, 30, so they'll ask you for 100 minutes, 60 minutes, 80 minutes, so you should be able to do. You should be able to do. Is this clear? This is your general understanding, okay, this is your general understanding. So this is how they proved that DNA replicates in a semi-conservative manner, okay, semi-conservative manner. Don't worry, problems I'll make you do. After the, not this class, once I finish this like major chapters, I'll make you do the problems. Okay, let's do one PYQ, few PYQs. First question, in Hershey and Chase experiment, what finding led to the conclusion that DNA is the genetic material? You have to be quick, okay, you have to be quick. I have one more class, that's why. In Hershey and Chase experiment, okay, DNA, how did they prove that DNA was the genetic material? Option number 1, 2, 3, 4. Okay, this is directly from your NCERT. Okay, this PYQ also was asked but in a different pattern. Question was in a different pattern. Answer, waiting. Okay, 3. Some are telling 3. Okay, the presence of radioactivity in bacteria when, okay. Very good, it's option number 3, okay, it's option number 3 that proved that the presence of your bacteria when P32, okay, the presence of radioactivity in bacteria when P32 was used. Okay, next question. DNA fragments are, this is NEET 2017 question, negatively charged, positively charged, neutral, depends on the size. Thank you, Madhumita. Madhumati. I always call her Madhumita. I think by now she's used to it. She's like, okay, she keeps changing my name, I'll just get used to it. Okay, tell me the answer for this. DNA fragments are negatively charged, positively charged, neutral, depends on the size. I think their chat's a little delayed. Here, yes. Okay, very good. 1. Perfect. The answer is 1. That is negatively charged. Okay, negatively charged. Next question. The final proof for DNA as genetic material comes from the experiment of NEET 2017 question. The final proof for DNA as the genetic material comes from the experiment of MacLeod and Avery, Griffith, Harvey, Hershey and Chase. Yes. Thank you, Divya, Savita, Jay. Yes, tell me the answer. Very good. Option number 4. Option number 4. Right? That is Hershey and Chase was the final proof that this was the experiment. Okay, next one. Homework question. Okay, 1 second before I go to the homework question. Students, next class, what and all we have to cover? We have to cover the enzymes involved in replication. Okay, we have to cover transcription, genetic code, translation, lac operon, and HGP and DNA. Next class will be little more longer than this. Today I did not have time, or else I would have taken for 1 more hour, but I have a batch class. Okay, so next class we will do the enzyme machinery. Okay, we have done up to experiment, enzyme machinery, and this class what and all did we do? We did up to Meselson and Stahl experiment. I will do a numerical next class if time permits. Okay, if time permits, we will do this. I hope everything is clear, students. There's no doubt, very important chapter. Okay, now ready for homework question? Okay, so after homework, you should also tell me how the class was in the comments. Okay, first homework question to all of you. Which of the following is not a component of nucleotide? Okay, which of the following is not a component of nucleotide? You have option nitrogenous base, phosphate group, ribose sugar, deoxyribose sugar. Okay, which is the part which is not a part of the nucleotide? Okay, nucleotide, nitrogenous base, phosphate group, ribose sugar, deoxyribose sugar. Yes, I have a DNA class. Okay, yes. Next question, two questions. Okay, two homework questions you have. Which of the following nitrogenous base is found only in RNA? Which is only found in RNA? Okay, adenine, thymine, uracil, guanine. Adenine, thymine, uracil, guanine. This is your question. Okay, so do the homework and let me know in the comments if genetic is easy with Gopika ma'am. So if you understood so far, comment below for part 2, that is genetics is easy with Gopika ma'am. Okay, my alarm rang. Okay, genetics is easy with Gopika ma'am, and definitely do both the homeworks. Okay, first question is what is a part of the nucleotide? You can write all the 3 components of the nucleotide, or according to the option you can tell the answer. Second question is which is not a part of your, I forgot, nitrogenous base. Okay, that is in DNA, which is the not part of your nitrogenous base. If you put the answer here, I'm not going to check. I'm going to check your comments and I'm going to like it from the main account. So students, let me know with this hashtag today, genetics is easy with Gopika ma'am, and thank you so much for joining. Awesome. Hats off to all the students who stayed from the starting from 10 a.m. and only if you comment, part 2 is going to come. Okay, because now we have to have that, right? We have to have that give and take policy. Giving, it's over, students. Not that big of a heart also. Okay, so yes, you should comment and let me know how the class was, because I'm taking, this is a botany syllabus. Okay, this is the botany topic. So thank you so much for joining. Tata, bye bye, see you, have lunch, take some rest, and see you soon. Ma'am will go to the next class.